juveniles. Joyce Williams.
It's getting scared now. It's getting cold.
Liberty Rose Lynn German.
Missing, abducted, we don't know for sure.
Last seen at the High Bridge in Delphi, Indiana.
Delphi is just not one of those places where any of us ever thought that something like this would happen.
Despite that video, despite that audio, nothing happens, seemingly, for 6 years.
The judge did find probable cause for an arrest of Richard Allen. He's been charged with 2 counts of murder for the murder of Abigail Williams and Liberty German.
Richard Allen, you child murderer! Why did you do it?
No, they got the wrong guy. There's no way.
There are other people who were looked at for this.
Shame on you!
It's some sort of a religious sect/cult
Shame on you, Richard!
that follow Odinism.
Shame on you, Richard Allen!
I believe they sacrificed a girl and killed another one.
And law enforcement insists no one's ever been cleared.
Baldwin, can you provide any comment?
Journalism. It's time for you guys to start being journalists.
Good evening, everybody. Welcome to Case by Case. I'm Amy. Welcome in everybody in chat. We are discussing tonight the DNA that was presented at the Richard Allen trial in the Delphi case. I have some bad news. Andrea Burkhart is right in the middle of an emergency. She is at the vet with her dog, having to put her dog down. It's very sad. She said she will make it up to everybody, but she's not going to be able to make it this evening. But we do have myself. We are accompanied by Cherie, who is an attorney from upstate New York. She does legal aid, and she also helps with preventing homelessness on the legal standpoint. Then we also have Christine, who's with us tonight. Christine is a paramedic from Southern California. She has worked as a first responder in Los Angeles Riverside, in Los Angeles Kern County, California, and she has worked for the Department of Homeland Security, providing medical support for detainees and customs at the Border Patrol. And I'm going to bring them both on. We have a huge presentation for you guys tonight regarding the DNA. And we're going to go through everything that Dr. Silkman has prepared for us. We're going to talk about, let me see, I have a list here. We're going to talk about DNA and the helix structure. We're going to talk about the genetic material found in the human cell. And we're going to talk about what biological sources DNA can be abstracted from. And we're also going to talk about hair follicle structures. We're going to talk about the crime scene. We're going to talk about the DNA that was collected. Everything that Dr. Silkman has based his presentation off of is from Andrea Burkhart's reporting from the trial. So I'm going to bring Christine and Cherie on now, and then we'll bring Dr. Silkman on, and I'm going to let him introduce himself, and we will take it from there. The chat is in slow mode, guys, and we'll get to questions later in the presentation. So let me bring him on now. Welcome, Dr. Silkman. How are you?
I'm much better, thank you very much. And I just want to say, first of all, I'm very sorry to hear about Andrea's situation. It happened to me as well. It's a terrible, terrible thing. But first of all, I'd like to welcome everyone that's on the panel and in chat. And guys, I'm truly honored to be here. I haven't been well myself for the last couple of months. And when Amy approached me a couple of weeks ago if I would do a DNA presentation on the Delphi case, I sort of said, oh, yeah, okay, I'll try it. And I never realized how hysterical and crazy this case is. So what I'm going to try and do is give a presentation about the DNA forensic evidence. I'm going to make it as respectful as possible because I understand the situation, but When I start the presentation, I'll introduce myself for those who don't know who I am. So back to you, Amy.
Okay, I'm going to pull up your presentation. And
Awesome.
It's a DNA forensic analysis, evidence analysis of the Delphi murder trial in 2024. And we're going to get into your biography and who you are and all of the credentials that you bring to the table. So I'm going to let you take the stage. Go ahead.
Thank you. And again, thank you so much for inviting me. I'm truly honored. And thank you, everyone in chat. I hope that you find this information helpful. Now, a lot of people know who I am. I'm one of the co-founders of the Foul Play team. And we have been working exclusively on the Stephen Avery and Brendan Dassey cases. When Amy told me about the Delphi murders, I started doing a lot of reading and research, and I was shocked to hear about the similarity between the two cases. But first of all, for those who don't know who I am, I'm from Adelaide, South Australia. So I'm from the land from down under, and welcome if there's any fellow Aussies here. This is my picture sitting down when I used to have a little bit of hair, and I'm sitting down with my fellow colleagues from the Department of Biochemistry. And if I could just say, the Department of Biochemistry back then had a center of excellence in molecular biology and recombinant DNA technology. So you can see that there's a lot of people here, professors, associate professors, postdocs, PhD students, honors students, general staff. There was a great big emphasis on molecular biological techniques. This was the 80s, and I was part of it, and I was so blessed to have some incredibly smart cookies and colleagues. You can see on the picture on my right-hand side, that's me receiving my doctorate, my PhD, and it was a real proud moment for me. Okay, so what are my professional and educational qualifications? Because I'm here, I'm giving a presentation on DNA. So I have a Bachelor of Science degree where I majored in both biochemistry and genetics. I have a first-class honours degree in biochemistry, and I have a doctorate, my PhD, in molecular genetics. This particular field that I studied and did my dissertation was in human molecular genetics. I did my PhD at the Women's and Children's Hospital, and I was isolating polymorphic markers to chromosomal fragile sites. So I understand the technology. I did the technology that I'm going to be presenting today, and I've worked extensively in a professional DNA laboratory. After I finished my PhD, I did a postdoctoral research fellow. I did a 5-year term, again, working in a professional DNA in laboratory environment, handling DNA every day. So you get to know the ins and outs of DNA. And remember, back at this time, there was a lot of stuff happening in the human molecular biology field. My final job, which I did for over 20 years, I was very lucky, and I had an associate lecturer position where I taught science, biotechnology, and medicine. I also taught students in practicals, in molecular biology techniques and biochemistry. And it's really interesting that the techniques that the DNA analysts used, we actually did it as a second-year practical, right? Where students amplified up their own loci, ran their gels. So it's a procedure or procedures that I'm very, very familiar with. And finally, guys, I have 14 refereed publications in the field of molecular biology and recombinant DNA technology. All right, if we can have the next slide, Amy. Just wait for that to come up. So the way I'm going to present the structure, sorry, Amy, all good?
Yep, we're good.
Yep. So the way I'm going to present this presentation, the structure of this presentation, as you guys know, the judge did not allow cameras or recording equipment into the courtroom, which is a real shame. So hence, the primary information that I'll be providing to you today, looking at the DNA forensic analysis, comes from Andrea. Now, a lot of you guys know who Andrea is, and she's got a huge YouTube channel, very, very highly talented, very well respected. The impressive thing about Andrea, who was in the courtroom, is that she took very extensive notes. So I'll be compiling and distilling down that information into this presentation, and hopefully it'll be of benefit for you guys. I'm also going to be showing some crime scene illustrations. That I have no access to any crime scene photos. Remember, I'm trying to be as respectful as possible, but I'll be using crime scene illustrations that were supplied to me by Richard Allen researchers, and I want to thank you so much. They're excellent and very helpful. All right. Now, the good thing about this presentation is that no matter what true crime you follow, if it has DNA in it, all the information I'll be presenting to you today will be very, very useful for you. So I've tried to distill it down to very basic. I'm not going to try and do anything overly complicated. I don't want to lose any of you guys. So I'm going to define what DNA is, look at its structure, and have a look at the different types of DNA molecules that are present in human eukaryotic cells, cells that make us up. I'm going to explain where you can get different biological sources as forensic samples for DNA analysis. So when crime scene techs go out to a crime scene of a crime, what do they look for? What do they try and gather to do further analysis on? That's very, very important. Now, for the geeks out there, I'm going to go through what PCR is, the actual technique. Don't worry, I've simplified it down. And very, very important, right, Amy? Especially in this case, and that is the generation of DNA profiles, how they're generated, what they mean. A large portion of the presentation will be summarizing what the DNA analyst found and presented to you using diagrams. And all of this is going to be very relevant to the Delphi murder trial. All right, I'm going to outline some of the issues with the forensic DNA investigation, and there were plenty of issues. I was actually quite shocked. But when you think about it, not to be surprised. Finally, I'll give a conclusion as to what was found and possible future DNA testing. And at the end, if we've got time, I'd love to answer some of your questions about the DNA forensic analysis. Can we have the next slide, please?
Yes.
Awesome.
Can I just say, Dr. Silkman, you're amazing. You're a great teacher. You really are.
Oh, thank you very much. I don't even start.
You're a great teacher. Like you explain everything it's a complex conversation, but you do a very good job of making it very simplified. I love it. Thank you so much.
Yep. No, pleasure. And I hope everyone in chat understands what I'm going to talk about. So what is actually DNA, right? Because I know that the true crime genre is huge. I've been in it for probably close to 7 years myself. Actually, I started back in 1985 when I looked at the Kennedy assassination. But now let's focus on this. So DNA itself is deoxyribonucleic acid, and it is the carrier of genetic information. It's what makes up me, you, everybody in the panel, and everything that's alive. So DNA is very important, and it consists of highly specific sequences, and I'll talk about that in a second. DNA actually is a very simple molecule. It's made up of just 4 bases, A, G, C, and T. So if you focus on the diagram in the center, you can actually see the structure of DNA. And as you can see, C base pairs with G, A base pairs with T. Now, if you could remember that, guys, you're a molecular biologist already, okay? Because that principle of complementary base pairing is crucial for forensic DNA analysis. Now, the structure of DNA was established many, many years ago by Watson and Crick. In our cells, DNA exists as a double helix. And if you have a look at the diagram on the extreme right-hand side, you can see the molecular structure of a DNA molecule. Guys, that's how the DNA predominantly looks inside your own cells. Now, what is crucial is this: the ability to both detect and extract DNA at a crime scene is the cornerstone in modern forensic science analysis, right? So if you go to a crime scene, it's very important that you're able to detect samples that contain DNA and to extract that DNA from a cell or cells. So the question is this: okay, we know the basic structure of DNA, but where do you find DNA in a human cell? All right, Amy, if we can have the next slide. So, where is the genetic material? So, a crime scene analyst, a DNA technician, where do they find DNA from? Where do they extract it from? If you have a look at the central picture, this is a cartoon representation of a cell, a eukaryote cell, typically what you find in a human being. There are structures within the cell called organelles. And these organelles, there are two different types. Amy, can I just pause my presentation here?
Yeah.
I'm going to come back. Give me a couple of minutes. I need to take some medication because I can feel my voice going.
That's fine. That's fine. And I'm going to answer some questions in the chat because they're overloading.
Awesome. Give me a couple of minutes and I'll be back. Thank you.
Okay. So, good evening, everybody that's here.
Good evening, everyone. Thank you for being here.
Hi, Cherie. I think everybody's sick. Dr. Silkman's been sick. You sounded sick today when I talked to you, Cherie. But Dr. Silkman is like, I mean, his depth of knowledge is amazing. And he likes to give all of you the breakdown of, you know, how DNA works, what they're doing in the labs. And I believe he's got like over 20 years experience in the lab. And I know that as all of these exhibits come out, because I've already talked to Andrea a little bit about the exhibits, what I'll probably do is I'll have Dr. Silkman and Andrea on together again once we get these exhibits, because I know Dr. Silkman is waiting on a lot of these lab reports. He can tell us a lot more, especially about the PCR reports, which give you kind of the definitive. So like when you have a mixture and the mixtures, they're unidentified, that can be very complicated. But it can show you, and he'll probably get into that much later. We're going to be here for a while. He's very detailed. And but when we have mixtures like we have in this case, we've had in others, and if you've not followed the Stephen Avery, Brendan Dassey case, which was the Making a Murderer on Netflix, Stephen Avery is represented by Kathleen Zellner. It's a very complex case with a lot of law enforcement and governmental corruption, just like this. And Dr. Silkman and his team have done what we are now just on the precipice of doing. That is fighting tooth and nail to get access to exhibits, trial transcripts. Foul play has gone so far as to get 9 phone calls where people were calling in and giving tips. I mean, he has a great he has a great source of knowledge on how to go about doing that, and we may end up needing that in this case. So he's going to get into how it's calculated when you have a mixture. And we're going to get into the crime scene and where the DNA was located, especially the unidentified DNA, the hairs, the animal hairs. I really want to ask him about the animal hairs. And then everybody who emailed me questions or posted questions in the Facebook group or messaged me or posted on the community page, I've pulled all of your questions into a PowerPoint presentation, and we'll save that for the end. But going forward, like if you have a question, we're going to try to, Sharia is really good at capturing those throughout the chat, putting them in the private chat for us mods and me, and we will try to get as much answered as we possibly can.
I like how you threw in the me because I don't know how.
Yeah, like this right here. I don't know for sure if we can address the rootless hair DNA standards. That is a question that we'll ask. That is absolutely, that's what he's here for. He's fully, Dr. Silkman has been, he understands the concept of complicated cases with DNA and everybody questioning how law enforcement handled a case. So, he can definitely guide us on giving us answers on the ins and outs of why something was done, why it wasn't done, and what this or that means. So all of like a question like that, Lindsay, is a great question, and we'll pull that in towards the end after he gets through his presentation. So welcome back. There he is.
Thank you very much. I'm probably going to have to take a few breaks.
That's fine.
I'm losing my voice. So if we focus on the central diagram here, where we're looking at a human cell, you can see that in a cell, there are certain specific structures called organelles. The two organelles that I want you to focus on are the nucleus and the mitochondria. So if we focus on the nucleus and have a look at the right-hand side, you can see what is known as a human genome. And a human genome contains genomic DNA. So a nucleus contains genomic DNA, which is present on structures called chromosomes. Now, if you look on the right-hand side, you can see that we have two different types of chromosomes, autosomes and the sex chromosomes. We have 1 to 22, so 22 pairs of autosomes. You inherit half from your mother and half from your father. Together, that's known as a human genome. So the important thing is this: we have autosomes, but we also have the sex chromosomes. If you're a female, you have 2 copies of the X chromosome. If you're a male, you have a copy of an X and a Y chromosome. This, guys, is very important, especially in forensic analysis. So to break it down, in a nucleus, it contains genomic DNA, and it's something that you'll hear often. Someone who works with DNA, an analyst in a courtroom situation, talking about genomic DNA. However, in our cells, we have another very important organelle called the mitochondria. And mitochondria, if you look on the left-hand side, contains its own DNA. This is remarkable. The DNA is small and circular, and there are many, many copies in a mitochondria. So mitochondria contain their own DNA, referred to as mitochondrial DNA. Guys, this is going to be very crucial when we're looking at hair analysis, right? So the important thing about mitochondria is that they are maternally derived. Therefore, all the mitochondria in your body, they come from your mother. None of the mitochondria come from your father. So Why is this important? Because a cell contains genetic information. So both genomic DNA and mitochondrial DNA, they can be used in forensic DNA analysis. This is crucial, guys, if you want to solve crimes. So, guys, are we happy so far with that explanation? Not too complex?
Perfect.
All right. So the question is, all right, but where are we going to get our samples from? You're at a crime scene. Where are we going to get our samples, our forensic samples from? So if we have a look at the next diagram. Okay, and what you'll see is there are many different biological sources that can be extracted to give DNA. So at a crime scene, typically what a crime scene investigator would do is look for these particular biological fluids. They are blood. Now, here's the amazing thing. Mature red blood cells do not have a nucleus or mitochondria. You can't extract DNA from red blood cells. Blood contains white blood cells and other components. That's where a scientist will isolate DNA from. And when I was doing my PhD, I isolated genomic DNA from blood, thousands of them. So I've got a lot of experience in extracting genomic DNA from blood. Other samples that can be used to extract genomic DNA are semen, saliva, urine, hair, we'll talk about hair in a second, teeth, bone, and tissue samples, for example, muscle and skin. So there are many different biological sources that a crime scene investigator can find and extract DNA from. At a crime scene, what normally occurs is that a crime scene tech will either do swabbing, and you can actually see the swab up above. They can either do scrapings and also cuttings. And in this particular case, a lot of the clothing, there were many cuttings that were done to sample for DNA. So these things are normally done by a crime scene investigator. Now, I want you to know something very important here. Not all of these samples are created equally. And by that I mean that you can normally obtain very high levels of DNA from blood, semen, and saliva. The other samples normally have very low levels of DNA. And guess which gives a very low amount? Hair. All right. So there's a plethora of biological samples that a crime scene investigator can examine at a crime scene and then try and extract DNA from. All right, Amy, if we can have the next slide, please. So one of the elements that's discussed a lot in this case is hair, right? Hair samples that were found on the bodies of both Abby and Libby. So can we extract DNA from hair? And the answer is yes, we can. If you have a look at the diagram on the left-hand side, you see a cross-section of a hair. Right down the bottom of the hair structure contains a follicle bulb. The follicle bulb contains very active cells. Hence, if you have a hair that's got a follicle bulb, that means you can extract genomic DNA from that hair. However, many hairs do not contain a follicle bulb. And in the past, that was it. Party over. You could not extract anything from that hair. However, now, with very advanced techniques, and only a few laboratories can do this, they're able to extract mitochondrial DNA from hair that doesn't have a root or a follicle bulb. But this is something that is not routinely done, right? Not many laboratories do this. It has to be sent away to a special laboratory that can extract mitochondrial DNA. Why is mitochondrial DNA so good? Remember what I said? The mitochondria have got multiple copies of the mitochondrial DNA. So even though the hair doesn't have a follicle bulb, there should be some mitochondria there. So you should be able to extract mitochondrial DNA. So guys, yes, you can extract both genomic DNA and mitochondrial DNA from hair. All right. Can we have the next slide, please? Thank you. All right. So now you've gone to a crime scene. You've isolated DNA, but what are you going to do with it? And the answer is you use this incredible technique called the polymerase chain reaction. And no matter what true crime genre you're in, what case you're following, the DNA analysis will definitely be using PCR technology. The PCR is a very, very simple protocol, and it was invented by this guy down here on the right-hand side by Kary Mullis, who was a very out there type of guy, and he had this big dream, and he dreamed about PCR. He invented PCR, and he won the Nobel Prize 10 years later, which is truly remarkable. Every laboratory in the world would be doing PCR. Like I said to you, our second year students at university did the very same technique. I've done thousands of them myself. And no doubt, maybe some of you guys in chat, say yes or no, whether you've actually done PCR either in high school or at a university. So what is PCR? Why is it so good? Okay, so PCR is a molecular biology technique. And what it does, it amplifies, makes lots of copies, it's like a photocopier. It amplifies specific DNA sequences millions of times. So you may start off with a very small amount of DNA at a crime scene, but by using PCR, you amplify it. So you end up with millions and millions of copies. That's fantastic. How's it done? You use specific DNA primers that bind to DNA at highly specific spots, and you use a special enzyme known as a DNA polymerase. Back in the old days, I'm talking about in the early 80s, whenever you used to do a PCR, you normally do about 20 to 30 cycles. Each time you had to add more enzyme, because one of the steps in PCR is that you have to heat it to 100 degrees. Well, if you know anything, if you put an egg on a frying pan, you're going to destroy it, you're going to denature it. When you heat a sample at 100 degrees, you kill the enzyme. So you have to keep on adding it. Well, guess what? They discovered that there were bacteria, you know, those thermal vents in the bottom of the ocean that produce a lot of nutrients, and it's very, very hot. Those bacteria contain an enzyme, a DNA polymerase, that, guess what, is stable to heat. So all you need to do is to add the enzyme once. You mix it in that very little small tube that you see down below. You put it in a thermocycler, and you come back in 2 hours. We love PCR. Why? It's cappuccino friendly, right? You set up your PCR, put it in the machine, go into the city and have a coffee, come back, boom. You've got your amplified DNA samples. Truly, truly remarkable. All right. Why is PCR so good? And the reason why it's so good is because a single drop of blood or a single hair with a follicle bulb, that's got enough DNA for a PCR, which is remarkable. So when you go to a crime scene, you don't need a huge amount of material. Why? Because PCR will amplify it for you. But there's one problem. And it happened in this case, happened in the Delphi murder case. PCR is very, very sensitive. So if the operator is not careful, they can easily place their own cells inside the tube. And that's why in the Delphi murder case, correct me if I'm wrong, Amy and guys, a lot of the jury, there were people there that were really clued in and said, oh yeah, how do you validate your controls? And I thought, oh my God, someone knows about DNA. It's awesome. So you have to be very careful because PCR is extremely sensitive. It's both a strength and also a weakness. You have to be very, very careful. Now, PCR is not perfect, and it happened in this case as well. the success of PCR is affected by multiple factors. So it's not perfect. Things like low DNA levels, DNA that's been degraded, and we'll talk about that a bit later, and also inhibitors that are present in the sample, you won't get a PCR reaction. And guess what? Dirt is an unknown inhibitor of PCR. So if you've got any dirt in your sample, even though you try your best to clean it, it will kill the enzyme. It will not work. All right. But, okay, we've got our DNA. We've amplified it. So what? Scientists developed, and I was doing a lot of this work myself because I was isolating polymorphic markers back in the 80s. Well, scientists discovered that there are polymorphic markers, and we'll talk about it in a second, that vary in length, and they're spread right throughout our chromosomes. And what scientists discovered was you can use PCR with specific primers to generate what is known as an STR DNA profile. My God, brilliant. STR stands for short tandem repeat. Don't worry, I'll explain it. You can go to the shop and buy it as a kit. And all of these DNA forensic laboratories use these kits in order to generate a DNA profile. Very, very powerful technology. Amy, how are we going so far?
Wonderful.
Awesome.
Do we have, so when it comes to the PCR and this case, and I know that you've been hoping to get some of the reports that the lab submitted as exhibits, and I'm working on that. How does the testing, and maybe you have a plan down the further on in the presentation, because we did talk. I did see something in there about the animal hair.
Yes.
Is the testing that you're discussing right now, the PCR, is it a lot different when it comes to animal hair?
Yeah, it's different. It's different.
Okay.
These the kits that people use, they are specific for human DNA, right? But however, if you have an animal hair and you extract the DNA, there's nothing stopping you doing sequencing, right? Sequencing known genes and comparing those sequences to what's available in the database, because there are hundreds and thousands of DNA samples from humans to microorganisms that are part of a database. So if you sequence that, say you've got hair, you go, jeepers, is this human or animal? Plus, a forensic anthropologist or someone that deals with hair analysis should be able to determine the differences between a human hair and an animal hair. But if that animal hair contains DNA, you can sequence the DNA or amplify part of the DNA using specific primers and compare it to a database, right? Very easy to do.
So what I'm taking from that is that there was animal hair,
correct,
in this case, and we know that there were throughout the course of the investigation, there were several search warrants that were served where there were allegations of certain animals being tested. So, is what you're saying that if there's animal hair and they're able to decipher, yes, this is animal hair, there's a database that should basically specify to them what type of animal hair it is?
Yes. There are DNA databases that contain hundreds of thousands of entries, right? So if and this happened to me, when you're working in a laboratory and you discover a gene and you sequence that gene, you go, well, what's it similar to? So you can submit that sequence to a database and you get all the matches. And it'll say, oh, it's similar to a rabbit, similar to a donkey, and it'll give you the percentage match. So you can basically work out what animal you're dealing with. So it's a very, very powerful database. Yeah, scientists use it.
But we shouldn't be sitting here 8 years later, because
No.
this happened in 2017. We shouldn't be sitting here wondering what animal hair was at the crime scene.
I don't think they're interested. I don't think they're interested.
Because they, I mean,
Yep.
You're saying that they very easily they should know the answer.
Oh, yeah, absolutely. If the animal hair can look, there are experts who examine animal hairs, human hairs. They can do comparisons. They can look at the structure. Furthermore, if the hair contains a follicle bulb, you can extract DNA, you can extract RNA from it, you can do some sequencing analysis, you can put it to a database, you get all the matches, right? It's very easy to do. I've done it myself, right? But quite frankly, there's a lot of unknowns in the Delphi murder case. Excuse me. All right, so shall we continue?
Yeah.
Yes,
Do you need to
please.
take a break just so you can get a drink or something?
Well, I've got my drink.
Okay.
I just hope I haven't bored anybody. Okay, so now what we have here, this is very, very important. This is an example of an STR DNA profile, and the DNA analyst would have got a similar result to this. The proper name for this picture is an electropherogram. So what it shows you is a DNA profile, and I'll explain all of this. So what does this all mean? How does the DNA analyst look at this and decode it? It's actually very simple. So if you look at the top on the left-hand side, you'll see this thing called DAS1179. Well, what the hell is that? Well, that is a locus, and don't worry, I'll explain what a locus and an allele is. But what you notice, there are two peaks at D8S1179 locus. What the hell does that mean? Those two peaks are alleles, and in this case, you have the number 12 and 14. Because those alleles are different, that means that that individual is heterozygous at that locus. You inherit one allele from your mother, one allele from your father. Guys, we're cool so far? I haven't lost anybody? We're all good?
Still here. It's fascinating.
Awesome. Then if you go to the right-hand side, you'll see this thing called CSF1PO. That's a locus. And you can see that there is one peak, and it says 10. But remember what I said? You inherit an allele from your mother, you inherit an allele from your father. Therefore, this individual has got two copies of the allele 10, but you see it as one peak. This individual is homozygous. So now you can see there are different, we actually say loci that are amplified up, right? You can see them all on this electropherogram. So when a crime scene investigator, say, detects blood, semen, saliva, whatever, they amplify up the DNA using this kit, and they obtain a DNA profile. Now notice there's something called a melanogenin, right? Can you see it? So a melanogenin is a sex marker. That's very interesting. So here you can see the presence of an X and a Y chromosome. So that's a male, right? So if there was only one peak X, then that means that the sample that you extracted the DNA from and amplified up is from a female. So now, not only can we get a DNA profile, we can determine the sex. And at a crime scene, that's extremely important. Okay, so far so good?
I have a quick question. Can you Can you the heterozygous?
Heterozygous.
Can you give us a definition of what that is?
Yeah. So see the alleles. Remember how you inherit an allele from your mother and an allele from your father. What these locuses are, and I explain for what they are in the next slide. A locus, in fact, why don't we go to the next slide and I'll explain. It'll all sort itself out. So what do I mean by a locus? A locus is a specific region of DNA at a specific chromosome. That's it. So it's a piece of DNA on a chromosome at a specific location. That's a locus. Okay? So what is an allele? An allele is simply a variation of the DNA sequence at that locus. So we have what are known as polymorphic alleles, which means Amy, I don't recall, Streets of Bakersfield, me, everyone in chat, we differ from each other in the length and size of these alleles. And some smart cookie worked out, wait a minute, if we look at multiple locations on the human genome and we look for polymorphic alleles, that means they vary in the population, we can determine a unique profile that's as unique as your fingerprint. But we do it at a molecular level. You get it? So a locus is a specific region of DNA on a chromosome. Alleles are simply variations at that locus. Takes a little bit of time to understand. So we can now generate short tandem repeats. What does that mean? It is a simple sequence and it's repeated multiple times. So we vary in the number of copies of these repeats. And that's how we generate that profile. Now, here's the good thing. Every one of us on the panel and in chat has a unique STR profile, except if you're an identical twin or you happen to be a clone. So therefore, an analyst can generate a genetic fingerprint. This is very, very important in crime scene analysis. Amy, any questions?
So the allele, which is a variation of the specific chromosome strand, that would be like your hair color?
No.
Well,
Totally. That's you hit a really good question. Okay. Most of these regions that we amplify up, we say are non-coding DNA sites. They don't code for anything. So when you look at an STR DNA profile, we've got no idea about eye color, hair color, taste, skin color, nothing. These are sites that a scientist uses to generate a unique DNA profile. What you're referring to is SNP, single nucleotide polymorphism. That's completely different.
So the allele, the variation is way more specific than identifying physical attributes to a person.
Correct.
So an allele would be, other than what I personally want to know, an allele describes what about an individual? That's
nothing. All it says is that at that particular site, you've got this number of copies of that allele. That's it. So when you look at the entire number of loci that have been amplified up, the scientist can actually generate a unique DNA profile. Remember when I showed you all those loci that were amplified up? You either saw 2 peaks or 1 peak. So what a scientist does is generate a table of all the values and then uses that to compare to other samples. And the analyst asks the question, all right, I'll give you an example. So I isolate DNA from Richard Allen, right? He's your primary suspect. I generate a DNA profile from Richard Allen. So I've got his DNA profile. I then, at the crime scene, I get hundreds of forensic samples and I isolate DNA from those samples and I try to generate a DNA profile. I then ask the question, is Richard Allen's DNA in any of the test samples? And you do a computer comparison. Done.
And when the comparisons don't match and he's not there?
He's not there. He can be excluded. Don't worry, I'll go through the whole lot.
You're amazing. I love you to death. You're amazing.
No, not at all. Like I said, and I do apologize for some people in chat who are molecular biologists. I had extra slides that I took out because I knew it'll blow a couple of minds. So I'm trying to make it as simple as possible. Think of it as a genetic fingerprint. All right. So if we could just go back one more slide. I haven't.
Go back.
Go back one more
go
slide.
Back?
Yeah, go back one more slide. So,
Here?
No, advance one more. No, you're going backwards. You need to
backwards.
go forward.
Let me go forward. Okay.
Forward. That's it. Right there. No, one back.
Okay.
That's it.
There we go.
Yep. I forgot to say something very important. And that is, if there are more than 2 alleles at a specific locus, it means you've got a mixed DNA profile, right? Because remember what I said, you're either homozygous or heterozygous, which means that the number of alleles at any particular location is 2. And unfortunately, if you get 3 or more, it means that you have a mixed DNA profile. That means that you've got multiple contributors, right? So what scientists do is this. If they get an STR DNA profile and they don't know who it belongs to, guess what they do? They upload it to CODIS. And CODIS stands for Combined DNA Index System. It contains millions of samples, right? America's got a huge DNA database. So if you're at a crime scene and you generate a DNA profile and you go, huh? Who the hell does this belong to? Did someone commit a crime? So what they do, what the DNA analyst does, is screen the CODIS database, and often you'll get a hit. So many crimes have been solved, many cold cases have been solved by getting a hit in CODIS. Say, oh my God, this individual committed the crime, right? All right, so far so good? So you can see that these technologies, the STR DNA profiles, are very, very powerful. All right, let's advance one more slide.
Okay, before you advance, and while we're on the subject of CODIS, can you explain who makes the decision? So you work in a lab, and you worked on criminal cases where you were processing DNA in criminal cases, and so let's take the Delphi case for example. When the expert for the state testified that she was provided like over 100 samples. So in the DNA lab, if you are provided that many samples, first of all, I'm sure protocol is different in every country, state, county, wherever. Yes. But if you're provided those samples, do those samples automatically get uploaded into the system? No. Can I ask why? Like,
Well, if you haven't committed a crime, there's no point in uploading them, right? So basically, and I'll explain exactly what the DNA analyst did. You only want to upload a sample that you don't know the origin of. So you get an STR DNA profile at a crime scene on a piece of forensic evidence, and you go, oh my God, it doesn't match any of the standards, right? And I'll explain all of that, and you go, who the hell does this DNA profile belong to? Then you upload it to CODIS and do a search. And so it's quite known that if you work in a laboratory, if you work for law enforcement, your profile is uploaded. Why? Because you may have accidentally touched something at a crime scene, left your DNA there, and so it's important for the analyst to remove you from the system, right? So if you've contaminated a sample, you want to make sure that, oh, okay, that's from me, right? However, there are many criminal cases cold cases, where they go back, they extract DNA, get a profile and go, who the hell does this belong to? So they need permission to upload it and search the criminal database, and you may get a hit, right? So you don't normally just upload samples to CODIS.
So if you let's say you are brought in as a suspect.
Yep.
And you're sitting in the interrogation room and you volunteer, you agree to give your DNA sample, and they take a buccal swab.
Yep.
Okay. And here we are, let's hypothetically say 6 months down the road, we have 20 buccal swabs from different suspects. None of those buccal swabs are uploaded into CODIS?
Well, I don't. Now, I'm not an attorney. Maybe I don't recall, would know. But I'm sure that each state has got a different law. Like, for example, some states in America, I heard that if you're charged and you have an offense, your DNA is taken and uploaded.
Well, I think, it varies from state, but I do know for a fact that if you are charged with and I believe it has to be a violent felony, in some states, you are immediately required to provide DNA. And then in other states, if you are charged with a violent felony and you do prison time, you are then required to provide your DNA upon release.
Yes.
So I wanted to, I actually made a note last night because I felt like it just completely correlated with what we were talking about here. Richard Buckland. Are you familiar with Richard Buckland? He's from the UK, and Richard Buckland was the very first male individual who worked within a vicinity of two women who were brutally sexually assaulted and strangled to death on walking trails within a 3-year span. Within a 3-year span, and he worked right within several yards of the two females were found within 3 years apart on walking trails. I think they were 100 yards apart, but 3 years apart.
Yes.
He worked in the same facility, and when they arrested him, he confessed to the second female, and he was ultimately exonerated.
Yes.
Because they were trying to basically prove that he had committed both murders, and he confessed to one, but he was adamant that he didn't confess. He didn't do the other one. But that was the very first DNA exoneration that we've ever seen. And I think it was in the early 80s is when it was.
Yes.
So the fact that we're dealing with all different types of scenarios with CODIS and, you know, where I think everybody in chat and everybody who watches criminal cases, especially when we hear someone testify, like the state's expert did, that she was provided 100 samples. People want to know, were those 100 samples, were there DNA profiles created for each one of those to test against the DNA that was found at the scene?
I'll explain. Yep.
Okay.
I'll explain all that. I'll explain all of that. I'll go
we'll
through
get to that. Okay. So I'll keep going.
Okay. So if we have, beautiful. So let's have now we're honing in to the crime scene itself. Okay. So, and I'll read it. Now, both
It's
Abby.
really quick. I just wanted to clarify in the presentation, the clothing that the girls had they're switched. You have Libby was the one that was nude. Abby had on Libby's clothes. So and here it's the other way around, but I think we've all kind of like miscombobulated the whole story, but go ahead.
Okay, so you correct me if I'm wrong, because I took these notes from Andrea. So both Abby and Libby had their throat or neck slashed with a murder weapon, and the murder weapon was believed to be either a knife or a box cutter. My understanding is that the weapon has not been recovered. Libby herself was naked, while Abby was fully dressed in some of Libby's clothes. Amy, is that your understanding? That the clothing that Abby had were from Libby?
Correct.
Okay, beautiful. Got that one right. Okay, yeah. So Abby's T-shirt, her jeans, and her jacket were discovered in a nearby creek. So, tree branches, and some of them were over 7 feet long, they were placed on top of their bodies in what appeared to be a V or cross-shaped pattern. Very interesting that an FBI agent had stated that the girls' bodies appeared to have been moved and staged at the crime scene. A very controversial piece of forensic evidence was the presence or discovery of an unspent bullet, .4 caliber round as a Winchester Smith & Wesson, that was found on the ground between the girls' bodies. And the girls' bodies, correct me if I'm wrong, but they were found on Ron Logan's property. Is that correct?
Yes, correct.
Beautiful. So now remember, guys, I'm not an expert in the Delphi murder case, and I do apologize. There's a huge amount of information to process and digest. I'm giving an overview, and I know that some of you on the panel and some people in chat know way more than what I do. My focus is looking at the DNA forensics. Okay, so here's the problem. If you actually go out to the crime scene, you could immediately tell there are huge problems, and that is, it's an outdoor scenario. It's out in the wild. And so why is this a big issue? Because DNA is very susceptible to damage. It can be damaged by UV light, the sun, chemicals, which are found in nature, heat, humidity, hydrolysis, which is water. And you can see there was water, there was creeks, there was moisture, and microbial activity. Bacteria, microorganisms, all will work together to destroy DNA, to degrade DNA. Plus, look at the environment. The environment is not pristine. It's a dirty environment. There's mud, there's dirt, there's plant matter. It's a disaster for DNA, right? So, trying to recover quality DNA from that type of environment, from that type of crime scene, is extremely challenging. Guys, did everyone understand that? Amy, everybody understand that? That type of environment is very challenging to get clean DNA samples.
Yes, sir.
Okay, awesome. So when I worked in the lab, we're working in pristine conditions. When we isolate DNA, all of our equipment is clean. our starting material is clean. For a crime scene investigator, you're dealing with the wild. You're dealing with the environment. And so trying to isolate DNA from some items is very challenging. All right. So now we're now entering the last part of the presentation. Sorry that it's taken so long. Okay. So what did the how was the forensic analysis done? Well, the state's forensic analysis was Stacy Bozanovsky, and she worked for the Indiana State Police. As stated, she had received more than 100 items that were forensically examined. In a criminal case, that's a lot of items. I think in the Avery and Dassey cases, there were several hundred items that were forensically analyzed. This is a huge amount of work. So what does the scientist actually do? They look for a specific body fluid. They then do a presumptive test. Now, I want to say straight up front, a presumptive test is not confirmatory. It gives an indication that a particular solution may be present or a body fluid, but it's not confirmatory. You need to do an actual confirmatory test to prove that body fluid is present, especially if you're making big calls, right? So you look for a specific body fluid, you do a presumptive test, you prepare DNA. She also did trace DNA testing. So basically, if you touch an item, you potentially can leave your skin behind, skin cells. We shed skin, our hair, all the time. So scientists have worked out that you need a minimum of around about 25 skin cells before you can actually isolate DNA from that touched item. What Stacy did was she quantitated the DNA first. That's very important. So when she extracted the DNA, she determined, all right, how much have I got? Have I got very little? Have I got a lot? So you quantitate first so you know how much to do, how much to use for a PCR reaction. Because if you don't use enough DNA or if you use too much DNA, your PCR won't work. So you got to quantitate. That's awesome. What she did was she amplified 20 autosomal loci. I think that's what the FBI standard is. It can vary even up to 24. So she had amplified 20 autosomal loci in order to get an STR DNA profile. And what she did, and Amy had mentioned this, the very important point, she compares unknown test samples which are found at the crime scene. What she does is compare them to known standard DNA samples. So, Stacy would have prepared DNA profiles from Libby, from Abby, from Richard Allen, from family members, laboratory technicians, law enforcement officers. So, these are all placed in a computer. The results are then compared, right? So you've got your test, you've got your standards, you compare them. That's how it's done. Now, here's the problem. She found both single and multiple source DNA profiles, which meant that for some of the samples, you had multiple DNA contributors. That makes it very, very complex in order to analyze. But here's the good thing. A specific individual can either be included or excluded as a DNA contributor. So if you've got all these samples and you've got your suspect's DNA and you ask the question, is the suspect's DNA on any of these crime scene samples? If the suspect's DNA profile does not come up in any of those samples, you can exclude him. And I know this is going to be very, very controversial. So that way you can either include or exclude an individual as a DNA contributor. But remember what I said, there's a very hostile environment. So the quality of the sample, the presence of PCR artifacts, contaminants, they all affect the interpretation of the DNA results. Okay? So it's very important to rule out contamination. And one of the jurors, I think several of the jurors had asked, well, what do you do with your DNA samples? Do you lock them up? You test your solutions, right? So you have to exclude the possibility that a DNA tech has touched the sample, left their DNA in the sample, or a law enforcement officer has touched the sample. So ruling out contamination is crucial for vilification, right? Now, you know, and some of you are going to get very frustrated, some items, hairs, were not tested at all for various reasons. Amy, do you have a question? Anybody have a question?
With some of the reasons they didn't test some of these items, would it be financial? Are these tests expensive to run for a laboratory?
Absolutely. Absolutely. So a DNA analyst goes, okay, I've got hundreds of samples. I've got to prioritize what's important, what can be done later, what can be discarded. Now, you're going to be shocked, but a DNA analyst doesn't check all the stains. They may only test some of the stains. But they've got to keep the swabs because a defender, the defense attorneys might say, okay, I want all the swabs. I want to test them myself. So a laboratory has to keep all the samples for further testing and for verification, right? But some of these tests, you need a specialized laboratory to do the test. And unfortunately, a lot of these tests are very expensive. All right.
So oh my God, I have multiple questions. So first of all, when you say that a lab tech doesn't test all the stains,
yes,
that's where, first of all, can you explain who makes the decision what stains are tested?
Well, isn't that interesting? Because Stacey was asked all these questions. She goes, that's not my call. So in other words, the investigator in the case will say, I want you to focus on this. Don't worry about that. Okay, we got these hairs, but I want you to focus on these items here, right? So say you're at a crime scene and you might find pools of blood, hundreds of droplets of blood. You might go, I'll test a subset. I'll test 5 or 6 because you're thinking it's likely from the same individual, right? So the investigator or investigators, they're the ones that call the shots. Stacey is the technician. She's the one that goes, okay, I'll test these samples. I'll test them for DNA. I'll do presumptive tests. I've collected all the samples, the forensic samples. One day, the defense will come along and say, no. We want everything. We're going to retest everything. So therefore, a lab has to keep all the samples, especially the clothing. And I know I'm going to make a big call here.
So,
Amy.
So when you say that the detective makes all of the calls, so when she's presented, or the investigator, when she's presented with a pair of jeans, does the investigator detective dictate to her, I want the button, the zipper, the waistband tested? Who makes those calls? Where an item is tested?
Yeah, these are fairly standard tests. So you have to ask the question, all right, where's the greatest chance I'm going to find DNA, right? Mm-hmm. So you think about it, a pair of jeans is big. You could potentially do 1,000 swabs, right? So what you're asking is, where is the likely region that you're going to detect DNA from? And that's why later on I'm going to talk about further testing that can be done. So you make that decision. So, for example, there have been a lot of cases whereby they've taken a shirt and they've cut it up into 50 pieces. Mm-hmm. And they everything because they're looking for DNA or stains. In another case, they might say, oh, look, I might just test the sleeves. I might test the collar, I might test where the buttons are, so they make a judgment call.
So
Remember
when
what
you say they make a judgment call, is that you as the lab tech making the judgment call, or is it a combination of both?
Probably both. And remember what I
Okay.
said, they're looking for stains, right? They're looking for stains. So if they go, oh, there's this reddish stain here, I'll do a presumptive test. Oh my God, it's blood. Therefore, I swab it, I test it, I extract DNA. Correct? Whereas if a shirt, area of a shirt is absolutely crystal clear, you go, there's nothing there. So I'm not going to test that. So they're looking for stains. Remember I showed you the list? The analyst is going, all right, where is the greatest chance of recovering DNA? So that's what they do.
So this is probably going to be a really complicated question, but saliva is a good source of DNA, right?
Huge.
Okay. So hypothetically, let's say you have a victim who's standing in front of someone and you've got two suspects in front of her and they're both yelling and screaming at her and they are exasperating saliva as they're yelling, okay? And these things kind of like sprinkle over the front of the shirt and you really can't see it as a stain. So if you take a buccal swab or a lab tech was to swab, say, when you swab an area on a shirt where you don't see a stain, what is the parameter of swabbing? Like, is it a swipe? Is it a small little spot? And then once you do, and you, let's say you get two different individuals.
Yep.
What is the protocol for that type of a swab on a shirt or even the skin?
Well, if you notice what Stacy did to a lot of the clothing, she cut up. She did cuttings.
Mm-hmm.
So what you do is isolate where the stain is, so you don't
What if
contaminate.
there's not a stain, but you want to test? Is there a standard for a size?
No, it depends on really what the stain is, right? Some of the stains can be very, very small droplets. Others are much more extensive. So remember, in a laboratory, they examine clothing using alternative light sources. They're looking for fluorescence. So even though your eyes don't see it, by using an alternative light source, they can detect for the presence of stains. Right?
So they would use that light source before they ever cut
a
piece of material to say, okay, well, there's nothing here, but we see something here, but you can't see it in plain light, but we see it. Okay. And then we're going to cut it. Okay.
Correct. So they'll do cuttings, then they do an extraction. For example, in the Stephen Avery case, there was blood on a seat in the car. So what Cherie Cohen did was not a swab. She actually cut a circle. She actually cut the fabric and took the DNA from that cutting. That's exactly what they did here.
Okay.
Right? Yeah.
Yep.
All right.
Okay.
So let's move on because we're almost finished. We're now getting down. So here's the forensic DNA analysis that was done. So, and I'm going to try and be as sensitive as possible because I know this is very, very tough. So sexual assault kits were done for both Abby and Libby. Now, my understanding is that no seminal material was present on the external body swabs. Vaginal, cervical swabs were done, and there was the possibility of seminal material. But remember, Stacy did a presumptive test. Presumptive test is not definitory. Also, she was not able to find any male DNA. External genital swabs were done and fingernail scrapings. That's standard. They look for DNA under the victim's fingernails and they extract it. They did that both for Abby and Libby and they found some male DNA, right? And that's interesting in itself. However, no DNA profile was able to be extracted. They couldn't get a DNA profile. There was insufficient amount for testing. So that meant that there would have been very, very low amounts. Now you think about it, right? You're being attacked and you claw at somebody, you can scrape off a lot of their skin underneath your nails. You normally would extract a lot of DNA. However, in this particular instance, there was no DNA profile. There was insufficient amount for testing. Now, a big deal was made in the courtroom. They didn't know where this male DNA had come from, right? And so the suggestion was made that, okay, if you mix your clothing with other family members, and this is true, it's been documented, you mix your undergarments, your underwear with other people's undergarments and shirts and pants, socks, you'll transfer DNA. Even if you've got laundry, powder, washing powder, the DNA survives. In fact, I think studies have shown that you can wash the clothing 3 or 4 times. Yes, you decrease the amount of DNA you get, but the DNA is still there. So you can transfer DNA when you're doing your clothing and when you're doing your laundry. I bet you now no one's going to do mixed washing, right? So it's a possibility of transferring DNA from one piece of clothing to somebody else. Also, there's DNA in the environment. The girls are touching things, whatever. You can get male DNA, female DNA onto yourself, onto your clothing. What I did find very interesting is that there was no evidence of any sexual assault. I'm not saying it didn't happen, but when the girls' bodies were examined by a pathologist, there was no evidence of any form of sexual assault or defensive wounds for either Abby or Libby.
Can I ask you a question?
Yeah.
Do you have a lot of extensive experience with the rape kit or the sexual assault kits?
Definitely
to know what the standard is to determine whether there was a sexual assault?
No. And
okay.
again, I'm the type of person that I stay in my lane.
Mm-hmm.
If I don't know something and I have no expertise or knowledge, I'll say no way. Something like this, something very important like this, will only come from the person, the pathologist, who deals with this every day, all day for years. Only they are able to tell. But that's why we need to get the reports. But my understanding is there was no evidence of a sexual assault.
No, there was not.
Or defensive ones.
Defensive ones. Right.
Yeah,
that's
really unbelievable.
Yeah, it is. So when we take the results on the forefront of what the expert for the state testified to with possible seminal material, it's a presumptive test. So that how does the lab determine that there's possible seminal material? Is it there or is it not there?
I've covered that as well. It's coming up in the
okay. Because I know that's a huge question in this case.
Yep.
And so then my other question is, when we talk about the transfer of the DNA with the male individual in like laundry and things like that. So when they do, say, fingernail swabs or they do the vaginal cervical swabs, and there's possible seminal material, and then under the fingernails, they have an unidentified. Are they able to, even though they can't give you an entire DNA profile, are they able to say that the individual under each separate sets of girls' fingernails are the same? Or is it undefinable that they can't even say?
Well, here's the problem, and that's why I've got the questions down below. I haven't seen the original reports. Now, when she says, there was some male DNA, did they only just find a Y chromosome or evidence of a Y chromosome, or do they have a partial profile? I bet you they only just check for the male Y chromosome I don't even think they've got a profile, right? Because if they had a profile, even a partial profile is enough to exclude Richard Allen or anybody else. But I haven't seen the reports. That's why I can't wait to get my hands on the reports, because that way we can be definitive, right? But here's the important thing. The laboratory policy is not to test for female DNA. They only test for male DNA. And Stacey was asked, what the hell did you do that? And that's because the investigator said, we believe the perpetrator is a male. And you're thinking, wait a minute, you're meant to be unbiased. You can't have tunnel vision. You're not meant to know any outcome. You're meant to just get the results and forward them, right? So it's interesting that they weren't testing for female DNA. They were testing for male DNA. So here are three. Yeah.
In almost, well, let's say 7 years. In 7 years, they at some point uncovered that there was unidentifiable female hairs.
Yes.
And then they never, what is your opinion about the fact that they never changed course? Or they never said, hey.
Because they were focused on Bridge Guy.
Yeah, but we can focus on Bridge Guy, but at the same time, we have these unidentified female hairs. So would in any lab that you ever worked at, would an investigator ever come back and say, hey, let's try to run this and look at that, if they were truly trying to get answers? They wouldn't do that?
No. I'm not saying they wouldn't do that. In this case, they weren't really that interested until it became very controversial with the hair that was in Libby's hand. Then they had...
Yes.
Abby. So then they had to find out, well, who the hell does this hair belong to? They were hoping it belonged to, guess who?
The sister.
No.
Yeah, they were hoping that, but yeah.
Right. So you got to understand, when you do a criminal trial, you got to present the client, your suspect, in the worst possible light. You can't produce exculpatory evidence that makes you look bad, right? So therefore, that's why certain items will not be tested and say, well, the lab policy really isn't there to test female DNA, right? We believe that the perpetrator is male. But then again, how did they know that the hair belonged to Kelsey, right? So they had to extract the DNA, do a profile, get a standard, and then do the comparison. I can't believe they waited that long to do it. Okay. But the audience is probably going, holy hell, seminal fluid? What the hell are you talking about? So how is seminal fluid detected? And how can you have seminal fluid but no sperm cells? Let me tell you now, if in that seminal fluid they found sperm, they would have found a huge amount of DNA, but they didn't. So now we've got this bizarre situation where the crime lab is saying, well, we think we found seminal fluid, but we can't get a DNA profile. There's something not right. So, and my other question is, how is male DNA detected? So I did some reading, some research. Seminal fluid is a very complex mixture. There are a lot of components in seminal fluid, and I'll read them. There's spermatozoa, which is sperm. And remember what I said? Sperm has got DNA, chromosomes, half the chromosome set that you inherit from your father. The sperm is full of DNA, right? So an ejaculate contains several 100 million sperm. If the sperm is there, she would have detected DNA. She found none. There was no DNA profile. She's saying that I found potentially seminal fluid. Okay, there's spermatozoa, there's seminal plasma, there's fructose, which is a sugar, there's prostaglandins, there are clotting proteins, there are flavins, there are amino acids, there's citrate, prostate-specific antigen. Most males will know about PSA because they go for a prostate test. There's acid phosphatase, amylase, zinc, alkaline mucus. So there are a huge number of components in seminal fluid. So what the hell did Stacey test? If we look at the next slide, Amy, you can see that there's a whole variety of tests that can be done to detect seminal fluid. There's the acid phosphatase test, using alternative light sources to check for the presence of sperm, that they can use an alternative light source, and they also do prostate-specific antigen. But guess what, guys? The trouble with the presumptive tests is that they can generate false positives. So even though you think you got seminal fluid, you may not have seminal fluid at all, right? There's something bizarre. You're saying, hey, I've got seminal fluid, but I can't get a DNA profile. So what the hell's going on? There are confirmatory tests. There's the Christmas tree stain, which is a color test for sperm. There's an RSID test for semen, which is a rapid stain identification. And the obvious thing to do is stick it under a bloody microscope, right? If you think you got sperm there, you take a sample, put it under a microscope, and you go, oh my God, I see sperm. Correct? So there are multiple confirmatory tests to prove the presence of sperm. But now the state is in this bizarre situation where they're going, I think there's seminal fluid. There's no evidence of any sexual assault. We can't get a DNA profile. What the hell's going on? So if you look at the next slide, here are examples in which a male can produce seminal fluid but no sperm. There are conditions, medical conditions. One of them is called obstructive azoospermia, which means there's a blockage in the tubes that carry the sperm from the testes to the penis. So you're not going to produce any sperm or very, very little sperm. This one is controversial.
Is that a medical condition, or is that a vasectomy? It's not a vasectomy. Okay, it's a medical condition. Okay.
Vasectomy is when you choose to have obstructive azoospermia, right? Because you cut the tubes. You see here, intentionally causes obstructive azoospermia, which means that if you cut the tubes, you can still produce seminal fluid, but you can't produce any sperm, right? The other one is non-obstructive azoospermia. The testes themselves do not produce enough sperm. And there's finally retrograde ejaculation. Instead of the sperm coming out of the penis, it goes into the bladder. So I don't think any of these were discussed during the trial, but I'm sure that in a post-conviction, they're going to ask the question, well, do you know whether Richard Allen had a vasectomy? Did he have any of these conditions?
I'm going to drop the bomb, and I have confirmed through direct sources from his wife that Richard Allen has not had a vasectomy. He has not had a vasectomy.
Yeah.
So
interesting. Very, very interesting. So now the state has to try and explain, well, maybe I didn't detect seminal fluid, right?
So, okay, so let me ask you this. So when they do the rape kit and they say on both girls that there's a presumptive positive for seminal fluid, if it's not seminal fluid, what is the alternative?
Females produce their own secretions, right?
Okay.
And that's why what you need is a physiologist to look at a female, a male, compare, because all the reading I was doing, the female secretions, you can get it wrong and think you got seminal fluid, but you don't, right? So again, you really need a physiologist to sort of say, look, at this age, these are the secretions that a female produces.
At 13 and 14, they secrete those?
Yes, I was
That's
going
what
to say.
Yeah.
At 13 and 14, I'm not going to get personal, but yes, and I have girls, and yes, that's the age where it starts to happen, and it's actually quite heavy at that age because of the hormonal changes that they're
Correct.
having.
Correct.
So, okay, so it could be either or, but in this case, with this lab, with this lab tech, was there a way definitively? I know that you went over the different tests that she could have done. Is there something that she could have? Can you go back over that? What
It's
she
so
could have
easy.
done to definitively say what it was?
Get the sample and put it under a microscope. That's it. And there's a whole heap of stains that you can do that stain sperm, right? You can't miss it.
Okay.
If the sperm are there, remember, a human ejaculate contains hundreds of millions of these buggers. So if the sperm is there, you'll be able to pick it up. I don't think she did that. Otherwise, she would have said it in court. Oh, by the way, I found sperm. You did? Did you isolate any DNA? No. No one's going to believe it. So she could have done all these confirmatory tests to say, yeah, it's highly likely that sperm is present. And that's
So
why
if she put it under a microscope, and the individual had a vasectomy and there was no sperm, what would she have been able to see under the microscope?
Sperm.
And what could sperm?
No sperm.
No sperm.
Right? Because with the vasectomy, the sperm aren't going to go out.
Right. So under the microscope, what would she have been able to determine at that point? Yes, this is seminal fluid.
Well,
versus excretions from a teenage girl.
That's it. That's what Streets of Bakersfield was sort of saying. And from what I was reading, females produce a lot of the similar chemicals that what a male does, right?
Okay.
And that's you want to do a definitive test rather than a presumptive test.
The
right?
estrogen levels in a female at that age cause them to have some pretty severe vaginal secretions. I have done laundry of little girls, and that's about the age that it starts.
Yeah, they do. So
Correct.
beyond the microscope, what would have been the quickest next step to determine whether it was seminal fluid? What could she have done?
Stain. There's specific stains that can be used as well. But I don't even know what presumptive test she used, because she said, well, we did a presumptive test. But the problem is there was no sperm, because if there was sperm, you would have got a huge amount of DNA. Plus, the examination of the girls did not indicate a sexual assault. So things are not stacking up. Right, because it was a presumptive test, the defense can argue, well, did you find any sperm? Have you got an explanation of why you would find seminal fluid but no sperm? How come you weren't able to get DNA? Right? So it's much more complex than that.
Okay.
But again, I'm not a physiologist. You really need to talk to someone who does physiology, who knows what occurs at certain ages in females and males. All right, now here's the kicker. Stacey was saying that they detected male DNA. The question is, did she actually get a profile or just test for the Y chromosome? So often you'll see people say, oh, we detected male DNA, but that could simply mean they detected the Y chromosome. The Y chromosome, and you can see the picture on the right-hand side, is specific to a male. So there are three basic methods to determine the presence of Y chromosomal DNA. One, through the use of short tandem repeats. Scientists have discovered that there are STRs which are exclusive on the Y chromosome. So you can incorporate this as part of your kit. Then there's SNPs, which are single nucleotide polymorphisms. Scientists discovered that there are specific SNPs which are only found on the Y chromosome. And the final one is what I had discussed. That was the amelogenin gene sequence, which I showed you in the profile. So there's a slight difference in the size of the X and Y gene, And so there's really 3 different ways you can detect Y chromosome DNA, right? So Y chromosome DNA is paternally derived, comes from your dad. All right. So again, I would love to get my hands on her reports so I can dig deeper. All right. So now, guys, we're at our final part of the presentation. So what I've done is I've read all of Andrea's notes, which were excellent. And I've used diagrams, being respectful, that was supplied by the Richard Allen researchers, and I've put it all together. So I've distilled it right down. So here are the findings. The grey sports bra, the black bra, the pink top, the Nike white shoe, all contained Abby's DNA. The iPhone was swabbed, and they found an unidentified mixture that couldn't get a proper profile on the iPhone. Remember what I showed you before? The iPhone where it was sitting on the ground. So you've got moisture, you've got the dirt. It's going to be very, very hard to get a profile from the iPhone. The Converse shoes contain Abby's DNA and a mixture of both Abby's and Libby's DNA. Depends on what the ratio was. Now remember, the crime scene detectives, they gather the clothing, they do chemical staining, they look for the presence of a biological fluid. So if they find blood, they do a swab, isolate the DNA and get a profile. Now that means that the analyst has got a standard of Abby, Libby, Richard Allen, and family members. So they've got DNA standards. So they can determine what DNA is present on the girls and their clothing. Guys, we're cool? So they've got their DNA standards. So there's no guessing. They know what they're looking for. Now, this is where it gets really interesting. Amy, did you have a comment?
No. I'm nope, I don't.
So this is where it gets interesting. So when they looked at the swimming sweatshirt, it had Abby's DNA, a mixture of Abby's and Libby's DNA, and lo and behold, they found DNA, male DNA, from a laboratory tech. And I believe they put that sequence, that unknown sequence, into CODIS, and boom, out came the laboratory tech. Also, on the jeans that Abby is wearing, they found Abby's DNA, Libby's DNA, and again, the same laboratory tech. Now, this raises a question. Is the male DNA that they detected on the other samples from the lab tech, right? Did they just look at the Y chromosome because they said, oh, there wasn't enough DNA to do a profile? You're wondering whether the laboratory tech had contaminated a lot of these samples. So that means that they isolated the full genomic STR profile of that lab tech, stuck it into CODIS, and boom, the name came up, right? So they knew that those samples were touched by the male laboratory tech. Also, they looked at Libby's body swabs, right? And unknown male DNA was found, but it was not enough to do a profile. So unfortunately, when it came to the DNA distribution on both the girls and their bodies and their clothes, Richard Allen's profile did not come up. Guys, would you agree with that, with those findings?
Yes.
And that means that whether they were able to create a standard for the unknown male DNA or not, they would have been able to exclude Richard Allen.
From here, yes.
Okay.
Absolutely.
So we've got, okay.
Yep. So the DNA, now remember, she did a whole heap of swabs and a lot of cuttings, right? So many parts of the clothing were checked. Was a bust. They did not find Richard Allen's DNA on any of the samples. Otherwise, that would have been the very first thing that would have come out in court. You, me, everybody in chat, we wouldn't be here. Agree? Had Richard Allen's DNA been found, we would not be doing this podcast here. It's as simple as that, right?
And correct. And then do you, based on your lab work and based on the testings that you've done in the past, looking at this diagram and where the descriptive unknown male DNA is, it's not specifically random. It's directly in certain specific places on both girls. What is your opinion? What is your opinion of that? Because I personally have a hard time believing that those specific regions of the body would be the only places that they're detecting male DNA just simply because they live in a home with a male and laundry's mixed.
Well, these are actual body swabs, right? They're actual body swabs. So that means that the DNA is on them, directly on them. Now, you don't know how that DNA got there, whether it was transferred from clothing or them touching themselves, right? You observe any young boy, young girl, they're touching themselves all the time. So therefore, how did that DNA get there? Was it through transfer, secondary transfer that can occur, or is that from the killer? But the DNA level was so low, they couldn't get a profile, right? So picking up male DNA, you can't comment on how that DNA got there. You know why? Because you don't have a robust profile. If you had a profile and it came back as either Richard Allen, case is over. But they obviously tried looking for his DNA. They couldn't find it. Furthermore, this unknown male, you don't know where it came from. You don't have a profile. There's no profile.
If they have the standard from the males who lived in the home,
yes,
this DNA doesn't match them either. Is that correct?
Well, here's the problem. I haven't seen any profiles. If I had the reports, I could tell you in 2 seconds. We can exclude or include Richard Allen in 2 seconds. I can look at the results and go, these are common alleles, or all the alleles are different. What are you talking about?
But in a case like this, that's now 8 years old.
Yeah.
Is there any reason that we wouldn't have a DNA standard from the males who live in the home? If there's a dad and a grandpa who lives in one home, and there's a dad and possibly a boyfriend who lives in the other home, after 8 years, and you've got unidentified male DNA, what would be the reason for us to not have a standard?
They don't have a profile. Amy, they don't have a profile. They just said male DNA. All they could have detected was the presence of the Y chromosome. They don't have a profile, right? If they had a profile, then we're cooking with gas. They don't have a profile. Otherwise,
when you say they don't,
she would have said it in court.
When you say they don't have a profile, does that mean, okay, let's say, for example, Mike Patty, Libby's grandpa, who lived in the house, they took his DNA.
Yep.
Okay, and they compared it to the DNA, or they have his standard. When you say they don't have a profile, what does that mean? If you have his DNA, that this is does this mean that it's an unidentified male? If it was compared, okay.
Correct, but I want to know how much of an STR profile they've got of this unknown male, and I bet you they got 0. They don't have anything. It said unknown male. What the hell does that mean? Remember what I just showed you?
Yeah.
Chromosome. All they might have is, my test showed the presence of a Y chromosome, but I can't get enough DNA to do a profile, so I just say it's an unknown male. You get it?
Yes.
So if they had a profile, they would have said, I generated an STR DNA profile from the body swabs of Libby. I don't know who that male is. I've got Richard Allen's standard. The unknown male and Richard don't match. They don't have a profile of the unknown male. They don't.
So this could be like on top of everything else. This could be the luckiest killer walking around.
Well, the crime scene itself doesn't make any sense at all. There's a lot of weird stuff going on in this particular crime scene. But again, guys, I'm not a criminologist, so I don't want to talk about stuff I don't know. But this crime scene worries me a real lot. So let's finish. So then they looked at the presence of hairs. So when they went to the crime scene, they were looking for the presence of hairs on the body and also on the clothing. And I've summarized it like this. So they found lots of hairs, I think close to 80. So they found many, many different hairs. Some of the hairs had roots, other hairs did not have any roots. So here's what they found. Hairs with or without roots on the jeans, the swimming sweatshirt, the right Nike shoe, the pink shirt, the black bra, the grey sports bra, the left Converse shoe did not produce a DNA profile. So it was uninformative. They couldn't get enough DNA. And that's it. They couldn't get a profile. Here's the problem. Most of the hairs that they found had insufficient DNA for testing. Now remember, it's not trivial to try and isolate DNA from a hair. It's very, very tricky. You've got a very small amount of material there. So it's not surprising that they couldn't recover much DNA at all. However, the hair in Abby's left hand that was wrapped around her finger produced the DNA profile that was consistent with Kelsey German. So the question is, what's the significance of that? Could the hair have come from the clothing, and when the shirt was placed on Abby, that somehow got caught on her finger? I do not know. Then they discovered animal hair. Now, let me get this straight. Does Ron Logan, does he look after animals? Has he got animals?
Yes.
Lots of them.
And why? So and when you said that they should have been able to take an animal hair and test it to decipher what type of animal it was. Yet
Correct.
we don't know.
No.
All we know is animal hair. They've
Correct.
They've never come out and said what kind. You're saying that they should be able to do that.
Should be able to do it for sure. And don't forget, animals are in the forest, right? They're rummaging around. It could have nothing to do with anybody. It could just simply be that the hair was present there. But still, for the sake of completeness, it should be tested, right? It should be checked. I 100% agree. Now, here's the head scratcher. There were 3 hairs that were not consistent with either Abby or Libby, but they were not tested. And the reason why they were not tested, according to Stacy, is, oh, no, we can't test these hairs. Why? Because we will consume the entire sample. So I can't believe it that they got these hairs. They don't match to Abby. They don't match to Libby. Why not test them? Who do they belong to?
Why
Right?
not why not test at least one of them to see where it leads?
Correct. So there are many gaps in the investigation that could be done, that should be done. All right, so let's move on. Here's the other complicating factor, and that is a lot of items were found in the creek. Now, remember what I said? The creek's got mud, it's got dirt, it's got soil. All of these things are a disaster for DNA. So I've summed it up like this. The tie-dye shirt had no DNA profiles. They couldn't isolate any DNA from that shirt. The Nike left shoe contained Abby's DNA. The grey sweatshirt had no DNA profiles. The underwear had Abby's DNA. Correct me if I'm wrong, but are there items of clothing that are missing, as in a bra and underwear?
I've heard it's panties and a sock. but I'm not 100% sure.
Okay. They recovered one pair of...
I've heard the same thing, that it was a pair of panties and a sock.
Okay. Interesting. So the underwear that they recovered had Abby's DNA. The blue jeans had a mixture of Abby's DNA and Libby's DNA.
And that, let me ask you a question. Okay, so Abby and Libby were absolutely two different size little girls. Libby was 200 pounds. Abby was, I believe, less than 100 pounds. They were so whatever genes that would be on Abby would never go on Libby. Now, you could put Libby's genes on Abby, but they would be massive. They're massively oversized. So the genes that were found in the water that they claim belong to Abby, how is it that, we would get Libby's DNA in the crotch of those genes?
Unknown. Remember, the clothing was in the water.
Right.
The water was a disaster because potentially you could get mixtures happening if the clothings were all bundled together. Potentially you could get transfer. Maybe. Now, those blue jeans, they belong to Abby. Is that correct?
The jeans in the water the blue jeans in the water belong to Abby, yes.
Right. And Abby had Libby's jeans on. Is that correct?
Yes.
When she was dressed up?
Yes. All right.
So somehow some of that DNA got transferred in the crotch area. How? I do not know. I've got no idea how that could have possibly.
I don't think anyone ever will know. And just like the questions of what Libby was wearing that day, because there's the huge controversial question about, was Libby wearing sweats or was she wearing jeans? And when you look at the crime scene photos and you look at what jeans Abby had on that were supposedly Libby's, they look a little loose, but they don't look like so overly sized. Yet there's DNA from both girls, and it's so very frustrating at every turn in this case.
Would DNA from both Abby and Libby in those jeans, what was it? Do we know what kind of DNA it was? Was it like vaginal secretions, or was it like skin DNA, or do we just know it was DNA?
DNA, I do not know.
Okay.
Whether they did a presumptive test first and said, I saw blood, I saw a secretion, whatever. It's unknown.
If it was a vaginal secretion, if it was two vaginal, or if it was Abby's vaginal secretion, would that indicate that she was redressed while alive? Because when you pass away, you're not still secreting, or are you?
That, again, that's something you need to ask a physiologist.
Okay.
I know that sometimes when you are deceased, there can be body secretions that can be released because all your muscle relax, so it's a different physiological state. And remember, the fear that both of these girls would have faced, encountering the killer or killers, it would have been horrendous. So it's not really sure how both girls' DNA had appeared in the one spot. I can't answer that question, but that's why when we can do further forensic testing, maybe we can find out a little bit more. So the bandana apparently was not tested at all. The pink sock, the black sock, and the black camisole had no DNA profiles on it, right? So the main problem is this. Because the clothing were in water and they were recovered from the creek, it means that you're going to affect the DNA. So trying to extract the DNA, trying to do a PCR profile is going to be impossible, just about impossible. Why? Because you've got the presence of mud, soil, and dirt. All right, we have a question from Trav.
Yeah, Travis is asking, why are they saying the 3 hairs with roots not tested would consume the entire sample?
I suppose because it's really a one-shot experiment. So if you've got one hair, let's think about this. You've got one hair. You've got to extract the DNA from the follicle bulb, which means you've got to destroy the hair, right? Because the hair has got keratin, very, very tough protein. You've got to destroy the hair to liberate the DNA, which means that while you're extracting that DNA, what happens if your experiment fails and you get no DNA or extremely low quantities is done? You see, that's why these hairs have got to be sent to a laboratory that routinely does it.
So let me ask you this. Prior to making the decision about testing these 3 hairs, does this particular lab tech, have the ability to determine whether or not, looking at these 3 hairs, if they are from the same, they don't match Abby and Libby, but do they match each other?
Unknown.
Yep. You don't know if she's able. You don't know if she would be able to decipher that.
There's a problem, and hair forensic analysis has been really regarded as junk science. The FBI don't do it anymore. They don't do hair comparisons unless it's a real obvious difference, like you got blonde hair compared to dark colored hair, or hair from an animal, hair from a human being, something you'd be able to easily tell. But the FBI don't do hair analysis anymore because trying to do hair comparison, it's a bit of a nightmare. But if they say that the hair doesn't match Libby and Abby, then there's some type of characteristic in the hair that allows her to discern that, hey, it doesn't belong to any of the 2 girls, right? So therefore, you would think that's your priority number 1. Did the killer leave hair behind? I don't even know whether they tested Richard Allen's hair, whether they plucked some of his hairs, pubic hair, and checked them and do a comparison. Because normally in a rape kit, they do a pubic hair combing, right, to check if there's any hair there and do a comparison. I do not know whether any of that was done.
So frustrating.
Yeah. All right. I got a couple of more slides and then we're done. Thank you so much. So they also did recover some hairs that were found on the items recovered from the creek. But again, unfortunately, it was a bust. They were not able to get any DNA profiles. And it goes to show that when you're dealing with a very hostile environment, an aqueous environment, it's going to be very, very hard, right? So ideally, if it was a perfect situation and you pluck somebody's hair, a couple of hairs, you've got plenty of samples, you can easily extract the genomic DNA or mitochondrial DNA. But trying to do this type of analysis in an aqueous environment where there's mud, dirt, plant material, unfortunately was a bust, right? I think the only thing that they obtained from the hairs was the fact that one of the hairs, or the hair that was in Abby's finger, was from Kelsey. Everything else appeared to be a bust. All right, now, nearly my final slide. This is looking at the actual crime scene, looking at the pools of blood that were present on the ground, on sticks, and also on the trees. And there's been a lot of controversy about the F tree, etc., etc. So essentially, what the investigators did, obviously, is noted the presence of lots of blood pools at the crime scene. I think it was Cicero, who was the blood spatter analysis and crime scene reconstructionist, was trying to work out what happened to both Abby and Libby, whether they were staged, whether they were moved, whether they were killed, etc., etc. That's completely out of my league. But I can easily report on the fact that DNA was isolated from the blood pools that were found on the ground, on the F tree, on some of the branches, and they came from Abby and Libby. What's interesting is this. If the killer had used a blade, it's unbelievable to think that the killer never cut himself, right? Never cut himself, never shed blood, never shed hair, never shed skin. because nothing was found of the killer, right? And this has led to many hypotheses of, well, maybe the girls were taken away, killed, and brought back. I do not know. So what a crime scene tech does, is they take a swab and they do a presumptive test. So when they're looking for blood, they can use, and I've listed it, phenolphthalein, leukomalachite, luminol, fluorescein, hemostax, to determine for the presence of blood, right? And I think there were many pools of blood, and a lot of the blood came from Libby. Now, correct me if I'm wrong, but is it true that Abby had no blood on her hands?
None. She was described as pristine. Like she had, I mean, I sent you a couple of the crime scene photos, and you can see you don't see any blood. You don't see any blood. The way she's positioned, she's kind of, in like a fighter position with her two hands in front of her elbows condensed to her chest. There's no blood.
And the expert who testified even said that it was one of the few cases she had seen where someone had been cut in the neck where there wasn't blood on the hands, because the natural human inclination is to reach up for the cut.
Mm-hmm.
And don't get me started on that.
I mean, even when you look at Libby herself in the crime scene photos, there's, to me, with the amount of violence to her neck, there's not enough blood. I mean, when the expert testified that she was upright and moving around after she was injured, and then she went into a sitting position, and then I'm assuming ultimately laid down, then she was moved. The front of her, and the only description that I resort to is if you go and, because they're very public, is the Nicole Brown Simpson crime scene photos, OJ's wife, and you look at her crime scene photos from where her throat was cut, and you go, what? There's no comparison here. I mean, there's no blood, even with the puddles or the areas around. And I mean, we'll never know until if they ever release the crime scene photos, which are sealed. But regardless of the amount of blood that they claim is around the area, her physically, the front of her, is not depictive of somebody who is bleeding profusely from the neck in an upright position, in my opinion. And I'm no one, but
the blood also goes up on the face instead of down. Like she has streaks of blood on her face, but they go up and not down. Like she was picked up and carried, or somehow
inverted. Like she was inverted.
Yeah. Again, guys, this is completely out of my domain, and I hate to say something wrong or controversial. That's why I'm just sticking to the DNA. But quite clearly, this is an area that needs to be investigated, right? Because here's what the state are saying links Richard Allen to the crime, and that is the unspent bullet cartridge that was found between the bodies of the girls. Now, apparently, there was insufficient DNA for testing. Now, the big controversy is the fact that, according to the analyst, Melissa Oberg, she was able to find these extractor marks on the cartridge and match the Sig Sauer P226 pistol that was owned by Richard Allen. And so Melissa Oberg is a tool mark examinist. I have no experiences with tool mark analysis, but with weapons. The only thing that I can say is that if it took Melissa Oberg 7 hours to explain her testimony and what she did, she's in big trouble. Big trouble. And the number one person that's going to go up, that the defense are going to go after, would be Melissa Oberg and the tool mark analysis. That's the only thing that allegedly links Richard Allen to the crime scene, that unspent bullet, right? But that's another podcast. That's another podcast with somebody who's an expert in dealing with firearms.
I can't believe that they were able to settle an entire case with the bullet casing on, like, I wouldn't call it junk science, but it's new science. It's not
It is junk
proven.
science. It is junk science, right? There's a lot of issues. And again, please, I'm not a gun expert, never claimed to be. But apparently what Melissa had to do was she didn't cycle the bullet in the 6 hours. She had to fire the bullet to get the extractor marks on the bullet. Well, whoever cycled that bullet didn't fire it, which means that what you're doing is you're altering what occurred at the crime scene. You're changing one of the parameters instead of cycling it and looking for the mark. And she said, oh yeah, I couldn't be definitive if I just cycled the bullet. However, if I fired the bullet, yeah, I reckon that the bullet matches the 6 hour that belongs to Richard Allen. And you're thinking to yourself, holy shit, this is now starting to get into voodoo science. So quite clearly, for the defense to mount a post-conviction appeal, you need to attack Melissa Oberg and her findings. And I bet you there'll be plenty of experts that will say, mate, you can't do this shit. What the hell are you doing? Right? So that's an obvious area to attack. All right. Nearly my final slide. So let me summarize. And again, I didn't know much about this case, but this gave me vibes of a case that I've spent a lot of time on. And here's the conclusions. And trust me, the DNA analyst looked at a lot of samples, did a lot of DNA preps. And there's one big problem, and that is no DNA from Richard Allen or actually anybody else was found at the crime scene or any item recovered from the crime scene. The state on DNA evidence cannot put Richard Allen at the crime scene. Despite testing all those samples, I can't put him there. So now that's why they've said, oh shit, what are we going to do? So they focused on the bullet.
Dr. Silkman, can I ask you a question? In all of your years of experience in cases that you've gone through, like in the lab, how often do you come across a case where you have this many samples tested and you don't come across an actual individual?
I can't answer that because I haven't worked in a forensics laboratory doing cases. I mean, I understand all the science, but there'll be people in chat that will say, oh yeah, but in this particular case, it was brutal, it was bloody, but there was no DNA of the killer. So someone will always find an example where they say, yeah, but in this case, there was violence, there was blood, but there was no DNA. But in this case, this is an up-close and personal crime in which the killer had to physically touch the victims. Agree?
Yeah.
The killers had to physically touch the victims because there's evidence that the girls were moved from one location to another. Furthermore, the killer or killers dressed one of the girls. So he was handling clothing. He was touching skin. And you think, okay, so he's up close and personal with the victims. And this, whoever it was, left none of their DNA. Was he wearing gloves? Was he in a hazmat suit? Lord knows. We don't know. We could only report what we found. So they didn't find Richard Allen's DNA at the crime scene or any of the items that were tested. Here's a big one. There was no serological connection. That is, they didn't find his semen, his blood, or saliva, or hair, right? From Richard Allen that's related to Abby or Libby, right? None whatsoever. Plus, yes, it was years later, they couldn't find any of Abby's or Libby's DNA on any items in Richard Allen's home either. So now we're left with a situation where we have.
So okay, let me stop you there, because that will be another question. So the vehicle that he was supposedly driving that day, the Ford Focus,
yeah,
and any clothing that he would have been wearing, that they claimed that they had retrieved during the search warrant, items that appeared to be similar to what was in the bridge guy photograph. If he didn't get rid of any of those items over the course of the 7 years, is there a great possibility that there would be no DNA of the girls?
Well,
in the car, especially in the car, because that would be a direct.
Well, here's the problem, right? DNA and forensic evidence can hide in small crevices that you've got no idea are there. So you think you may have cleaned your car properly, but spots of blood, other bodily fluids can hide, and they would have checked the car with an alternative light source. They would have checked everywhere, and they would have swabbed everywhere. And I think they also did a carpet cutting as well.
Yes, they did. Yeah, they did.
Blood soaks down to the bottom of the carpet. So you think you've cleaned it, but you haven't. It's still there. But just over time, especially if you're washing clothes, over time, you're going to eventually lose that DNA, right? But surely, if he has committed this crime, I can't see how he wasn't completely covered in blood from head to toe, and his clothing would have reeked with blood and other biological fluids. And again, I don't know the case well enough, but I think one of the witnesses said that she saw somebody who looked bloody and muddy. And you think, holy shit, that's got to be a killer, right? And I can't believe that no one had noticed either a change in Richard's demeanor, having killed 2 young girls for what? A change in his demeanor? The fact that he actually went to the police and said, yeah, I was there. I saw such and such, and then I went home. He actually came forward, right? And his wife or daughter or any work colleagues never picked anything strange in his behavior or demeanor. It's hard to reconcile. Can it happen? Yeah, but I don't know. Again, I'm not a criminologist. I do not know. So now we're left with these hairs. So what can they do? Well, there are laboratories in the US that could potentially isolate mitochondrial DNA from those hairs to determine the profile, where they come from. Remember, mitochondrial DNA comes from the mother, paternally and maternally derived. So potentially they can look at that. They can do SNP analysis. So potentially they can do genetic genealogy studies. But hey, what the hell do you need to do that for? You've got Richard Allen's DNA profile. You got it. You would only do SNP analysis if you get a DNA profile and you go, who the hell does that belong to? Right? So hence you would do SNP analysis to say, okay, are any family members in databases? Can we determine who left this sample here? Right? I think that's how they caught one of the killers. Was it the Golden State Killer that did SNP analysis, genealogy studies?
Yes.
And they also caught the Gilgo Beach guy from testing something off of a pizza crust.
Correct.
Yeah.
Correct. And so what they could do is outsource the root hairs, the hairs, and send it to a laboratory and get it tested. But the big question is, why didn't they test it there before, right? Because if you're in court and you go, ladies and gentlemen, these 3 hairs come from Richard Allen and it's got his DNA profile. Are we going to be here doing this podcast? No. It's party over. But the fact that they didn't means that they're scared and they're worried, because if those hairs were at the crime scene, if they get a profile and it doesn't match Richard Allen and it comes from a male, then they're going, oh my God, we've got an unknown male profile here. Who does it belong to? Right? So it's not going to look good for the state. Right? And so my second to final slide, when I saw all of this, when Amy put me onto the case, this is what you guys dealing with this case are going to be faced with. And that is, people in chat are going to come online and say, you're an idiot. Just because there's no evidence, it doesn't mean that it's not there, right? And trust me, I've spent years studying the Brendan Dassey, Stephen Avery case, and now this is what the state are saying for Brendan. There is zero forensic evidence. There's nothing that backs up his claim. They can't put him at a crime scene. They can't put the victim in the crime scene, but he's still in prison, and he's going to be, I think, close to 60 by the time he gets out, if they let him out. So you've got this now situation, a very interesting situation in the Delphi murders case, where the DNA forensic expert has done all this testing, forensic testing, hairs, clothing, fibers, DNA, swabs. They can't put Richard Allen there. So now they're going, oh yeah, but his bullet, that bullet matches the gun. Yeah, but how did you get that result? I had to fire the bullet. What the hell, right? So now, trust me, you got years of this to look forward to, because this case, the Delphi murder case, is like the Avery case 10 years prior. You get it? You guys are just starting the journey. And I hope that my presentation today was helpful. Sorry it took a little bit long, but the important thing in all of this, something that we mustn't lose focus in, that we must remember the two little girls, Abby and Libby, who lost their lives. And that,
Absolutely.
Again, guys, thank you very much. Thank you for listening.
So we do have some questions.
I have one question. How possible would it be for someone to commit a crime this intimate with no DNA exchange? Like, in your opinion, based on what you know about DNA and DNA transfer, like how hard, like what is the probability of that?
No, I would say it would be very, very low. Can it happen? Yes, it can. But often what happens is that criminals that commit these crimes clean up after themselves. But, okay, putting bodies in water will definitely have an effect. But then again, despite potentially if the bodies were in water, I don't know if it was proven that they were in water or not, the crime scene detectives still found DNA, right? There was plenty of DNA there. And the fact that there were pools of blood present meant that the girls were likely killed at that spot, not in the creek. You get it? So therefore, think about it. If Richard Allen had committed this, somehow he had to escape and evade of being caught and somehow get all the way home. And remember, you got blood spatter. You're cutting arteries and veins. You've got spurting of blood. It's going to be very tricky to think that whoever the killer was left no DNA. But here's the problem. They can't find anybody else's DNA. But here's the trick. Here's what I suggest. What they've got to do is MVAC the clothing. MVAC it. Don't do swabs, but MVAC. And why is that important? Because MVAC extracts, if there are any cells there, MVAC will extract them, which means that if the killer had left skin cells on the clothing, remember the clothing are tough and fibrous, they're abrasive. If the killer or killers have left their DNA on any of the articles of items of clothing. MVAC it, test it.
Would this lab have the ability to MVAC it?
No. Get rid of that lab. Get rid of the lab. What you do is you send it to a forensics laboratory that specializes in this technique. You know the bullet?
Mm-hmm.
Swab? You don't do that. What you do, I think there's a technique, Bertolli method, where you actually dunk the bullet in a solution and you extract as many cells as you can from the bullet. Then you do a DNA profile. You don't do a swab because the amount of cells you're going to get back is minuscule, right? So send it to a proper laboratory that does these techniques.
Okay. Thank you, Rochelle.
Thank you.
Okay, so we're going to get to some questions. Is that okay?
Yeah.
Okay, so this is for you. It was by Pepperpot 5480. They asked, question for Dr. Silkman, how can a DNA profile be strong enough to exclude Richard Allen, but not sufficient enough to create a full profile? Thanks from the UK.
Yeah, so basically when you're preparing DNA, what you want to make sure is that the quality of the DNA is very good. So when you do an STR DNA profile, you get a full genomic profile, right? At 20 loci. However, if the DNA is degraded, not all the loci amplify up, which means you get a partial profile. Now, here's the kicker. Even if you get a partial profile, can you exclude someone? Yes, you can. So say you've got, instead of 20 loci, you've got 10 that have come up. You've got Richard Allen's full DNA profile. You check the alleles that have come up on that partial profile, compare them to Richard Allen. If there are differences between the two, Richard Allen is excluded. So you don't have to have a full profile. That's why I want to see Stacy's results, because that way I'll be able to tell you whether there are alleles which are common or alleles which are different. Okay, thank you.
Okay, so this next question is for Andrea. It was for Andrea, but it's in the presentation. So maybe, we'll take a shot at it. After viewing the crime scene and autopsy photos, in your professional opinion, were the girls killed at that location?
I think Cicero said that he believes that the girls were killed at that location based on the blood, the pools of blood. So if you cut the neck and you got huge amount of bleeding, you're going to find the pools of blood on the ground, right? Now,
and then if you take into consideration the crime scene photos and you look at Libby and you see that there's not that amount of blood on the front of her,
here's a problem. You don't know how they were killed. You do, for example, I hate to be crude. For example, if there were two people, they could have grabbed the girls by the feet and strung them up and cut the neck, and the blood went straight down. And that will explain why some of the blood went up. You get it?
Mm-hmm.
So held up, upside down, and had their throats cut, or furthermore, if someone grabbed the hair of the girls and pushed their neck back and sliced their throat and then let go, then obviously they're going to stumble, they're losing blood, they're going to be unconscious, fall to the ground, and you're going to get the pools of blood. But again, you need a criminologist to put it all together, right? But then you're starting now getting into the territory of, shit, were there multiple people there that carried this?
Right, yeah.
Yep, correct.
Okay, so this next question is for you, Dr. Silkman. It says, when a lab tech says they have, and I just copied and pasted these, when a lab tech says they have X amount of swabs collected in a criminal case, who determines which swabs get tested? For one suspect DNA profile to be created, from a collected sample.
Yeah,
How long, and they're asking how long does that take? So
Yeah,
for you, so if they give you a swab,
Yeah.
how long does that take?
So normally, I'll answer the first part. Normally, the chief investigator will have a look at all the samples that are collected, where they've been collected. Now, what will be very interesting is if the crime scene was videotaped.
I think. Well, I don't know about videotaped. I know that they had like extensive photography that was done, but I don't know for sure about videotaping. But I know that they did the crime scene in layers. They photographed it in layers.
Okay. So that way they can. The investigator says, right, these samples here have top priority because we need to know whose blood, whose semen, whose urine, whose saliva, whose hair it belongs to. So the investigator will say, right, we'll give it priority. So it's the investigator, not the lab tech. The lab tech is, okay, you want these samples done? No problems. How long does it take? So if you have a swab, and we did this experiment in the lab, right? And it basically took a day, right?
Okay. That's actually a lot quicker than what I would have thought, but okay.
So basically, if you have a swab, and what we got the students to do was do a buccal swab, right? And so you do a DNA extraction that takes a couple of hours. Then you set up your PCR, and the PCR can take maybe 1 or 2 hours, depending on how many cycles you do. Then you take your sample, and depending on how you view the result, you run a gel. So maybe let's push it out 2 days max. Yeah.
Okay, so you could possibly do in a say an 8-hour shift where you have a lab tech working for 8 hours. You could possibly do 4 to 6 buccal swabs. You could cycle 4 to 6 and then come back in the next day and they'd be done.
Yeah.
Okay.
So essentially, but remember what the analyst did. She did extra steps. She quantitated the DNA. Now, a lot of these steps now are done by machines. It's all done by machinery, by computers, robots. So it's much more streamlined because remember, these laboratories are processing a lot of samples all the time. So if you were to do this with a machine, I don't know what the modern machines are, I dare say a day, right? There are machines now that will do the entire process. All you got to do is put in the sample and it'll generate everything for you. They're very expensive. So I would say, depending on how many samples you process, what type of sample it is, give it between 1 to 2 days to get a result.
Okay. Here's another question. Is it, and this was for Andrea, is it strange for the police to collect DNA on a suspect in an old case that they say they didn't have enough DNA to generate a profile? What did they plan on doing with Rick Allen's sample in terms of comparing it to what? I don't.
Yeah, I think the question being asked is, if you have your DNA taken and it's placed into CODIS, potentially what they can say is, I wonder if he's committed further crimes. Will we get a hit to other crimes, other old crimes where they've got DNA samples from unknown cases? They submit them saying, okay, we put the DNA in the database, this person is missing, etc., etc. Then what they could do is look to see if they get a hit. And what they find in a lot of cases, when they catch a criminal and they get their DNA and they put it into CODIS, sometimes they get multiple hits, right? Some of these killers have been killing for a long period of time. They've got a big list, especially serial killers, got a long list of victims. So that way they can potentially solve historical crimes. Yep.
So
but the legal aspect, I don't know.
And that's where I was saying earlier, you know, if someone concedes to giving their DNA, why would that not be put into the database? I mean, if you're saying take my DNA, test it, well, I don't understand, because I think eventually we're going to get to the point where everyone's DNA is going to be accounted for.
Oh, there's a lot of people are very worried about SNPs, because if any of your family have given a DNA sample and they've done SNP analysis, then potentially they can pattern match and do genealogical studies and potentially hone down certain individuals, right? So there's all these things to do with privacy, right? And so there's a lot of issues here. Yeah, but they're legal issues, correct?
Wow. I think that's where the big issue comes in. Like they're hearing that there was 100 samples turned over, but probably none of them were tested.
Yeah, it depends. Like I said, I would love to get my hands on the DNA forensic reports, because normally when I do a case, when somebody asks me to look at a case, that's exactly what I do. I look at all the testimony, I read all the testimony, go through the analyst results, look at all the reports and tally it all up, work it all out. But Andrea did a beautiful job. We're very lucky to have her.
So this next question is for Andrea, but it's for, it should be for you and her, based on Stephen Avery's case with the whole concept of evidence being preserved. What evidence is at risk following Gall's denial of the preservation of evidence request? So the defense requested that evidence in this case be preserved, and she denied it, which in most states, I believe there's a statute that states that that biological evidence, maybe you know more than I do, Dr. Silkman, but and I would think that there's a statute that says certain aspects of the case needs to be preserved. What they were asking for was for the evidence to be preserved in the Reese Davis letters that the state initially claimed they didn't have, and then presented in their response to a filing that they got like 3 of them, but the rest were missing. So the question is, what evidence is at risk following Gall's denial of the preservation of evidence request? And are there alternative avenues available to ensure potentially critical materials are not disposed of ahead of Allen's appeal? We'll start with that question because it's a multi-question.
Well, put it this way. In the Avery case, there are statutes in place that say forensic evidence has to be preserved. Biological evidence has to be preserved until either a person in prison dies or potentially is exonerated. However, the state of Wisconsin gave cremains back to the family, and they are denying Kathleen Zorn any access to the RAV4 or any other pieces of forensic evidence, because they know that if the defense attorneys gets access to the forensic evidence in the Delphi case, and they find another male or male profiles that the state didn't pick up, boom, you're saying, well, you can't find my client's DNA in there. He's sitting in prison. What about these 2 DNA samples? You missed it. We retested the clothing. We used MVAC. We used other techniques. We found them. What are you going to do about it? The state do not want the stuff to be retested at any cost.
No,
so if the judge says we're not going to preserve the forensic evidence, oh my God, that's not a good sign.
No, it's not. And correct me if I'm wrong, but and for those of you who are familiar with the Stephen Avery case, I believe it was the prosecutor himself who sat and help write the legislation about preserving evidence in criminal cases, and then turned around and
gave it away.
They have this burn pit and multiple other areas on property of all different types of bones that continuously popped up that weren't there originally, but they have bones that aren't human, that they have determined are, some might be human and some might be animal bones. And when Kathleen Zellner, which is Stephen Avery's his attorney, she requested that they be preserved, they gave them back to the family. So the family accepted possible human and animal bones from the state.
Somebody else.
Yeah, and someone else from the state. And the prosecutor for the state, aka like a Nick McClelland, the prosecutor sat on the board to implement the law that said certain evidence like this, biological evidence, had to be preserved. And they gave it all away. So this is why this case is so horrendous, because some of us have seen it before.
Yes.
We've seen it before, where the corruption and, like I've said, we will know how far this chain of corruption goes when we get to the appellate court and what they say. If we don't get any relief there, we know that,
But I think it's pretty clear in this particular case. And again, you have to be respectful because it involves the murder of 2 young children, which means
and you know what? I want to just point that out. I think that for everybody who believes that Richard Allen is guilty, I respect that. And I understand why you can think that. However, people who are invested in these types of cases, who dig deep, the ultimate reason we're here is because of Abby and Libby.
Correct.
The 2 little girls. And I think that everybody who supports Richard Allen, you're looking at it as support for a child killer. We're looking at it from not only a standpoint of we want justice, real justice for these 2 little girls. And I'm here to say, if it's Richard Allen, okay.
Yeah.
But the state didn't prove that. And
no,
they did not.
Didn't.
They did not.
We face the same issues with the Stephen Avery, Brendan Dassey cases. The amount of vitriol that we have received, and you guys are going to get it too. The blowback you're going to get is going to be phenomenal. And like I said, when I looked at this case, I'm there, oh my God, this is the Avery case, but 10 years prior, right? So you're really at the precipice. The amount of blowback that's going to occur. Well, you're supporting child killers. How dare you do this and how dare you do that? Then they're going to throw to you about, well, okay, there was no evidence. It doesn't mean it wasn't there. Okay, well, if the evidence is there, you haven't found it, let's go back. Let's do a deep dive. Let's do some further analysis. If Richard Allen's DNA is there, I'll say he's where he deserves to be, right? And that's the philosophy I've got with Stephen and Brendan Dassey. Right, especially with Brendan, because Brendan is a very, very similar case. And now, because I saw the confession videos of Richard Allen with the police, and I was looking at it, and I had to double look. Say, oh my God, are they interrogating Stephen Avery? No, it's Richard Allen. It was exactly the same.
Yeah.
The thing that they threw at Stephen and Brendan, they threw at Richard. Precisely. Their work, it's called the Reed technique. Andrea covered it beautifully in one of her presentations. It's the Reed technique. And it's like, oh my God, they're going to do this. They're going to ask this. And I even predicted what they were going to do, and they did. They got his family involved, because notice what they were trying to do they were trying to convince the wife and the daughter that Richard was the killer. So what they were trying to do is to turn family against him, and they do it strategically, right? And then the worst thing is he's permitted to lie and fabricate to Richard Allen, directly lie, as they did to Stephen. And Stephen's looking at the detective saying, what the hell are you talking about? And the same looks on Richard going, huh? What? What are you talking about? That they have an accusatory tone. Believe me, if you're in that interview room in front of the detectives, they already think you're guilty. They're already treating you as suspect number one. You're toast. That's how it works, the read technique.
Yeah.
Okay.
And when you are advocating for victims, you in a case like this, or many others, you're advocating also for someone who, Stephen Avery, Brendan Dassey, Richard Allen, who you believe have been wrongfully convicted. It's such a depravity of what you would believe our justice system stands for, and it's sickening. It's sickening and it's sad. And when you say justice reform, and I talk justice reform, what I'm talking about is the fact that you have a process. There's like this, you have a speedy trial possibility, you have defense attorneys, you have a prosecution, but once you are convicted, your life is no longer yours at all. And when and in this case specifically, Stephen Avery's case was a little different. He was held in custody, but he was not sent to a prison. Richard Allen was treated like the worst criminal in the world and injected with medication that he didn't want, was treated like he was a child killer before he ever even stepped foot in the courtroom. That is not how this country and our law system is supposed to work.
He had no presumption of innocence.
None.
Same with Brendan when Mr. Kratz did the press conference. And unfortunately, I see the similarities in this case. You've got the death of 2 young children. You've got a sheriff heavily invested in the case. You've got a community in panic. Oh my God, is there a killer, a serial killer, child killer? What the hell's going on here? They need to solve the case. So hence, they are pattern matching, right? So no one, as far as I'm aware, has come forward when they originally saw the bridge guy and say, that's Richard Allen. What are you talking about?
No one did. No one did.
And so now the police are going, yeah, but you said you're on the bridge. Because I told you that. I told you that years ago. You put out some type of report that said, if you were there, come forward and tell us, we're treating you like a potential witness. He came forward, but no one said, I think it's this guy. He looks exactly like.
And the thing is that there's only one thing that would have made that whole entire interaction with Dan Doolin and Richard Allen not be significant, and that is the time of the day that Richard said he was there. And then years later, with shredded reports, no video interview, which Dan Doolin interviewed everybody with a video. Now we're supposed to believe 7 years later, during an election season, that, we just overlooked it. No.
Yeah, there are many grounds of appeal here.
There are. And there's been hundreds of grounds of appeal in the Stephen Avery case too.
Yes.
And they've all been denied, and that is what is scary.
the state, you think about it, after all that Richard has gone through, say he is innocent, after all he has gone through, his family, his daughter, his reputation is destroyed for several generations. How can you recompense him if he is innocent? You cannot. So hence, what the state have to do is put a lid on it and deny. Because once you open that lid and Pandora's box comes out, oh my God. And don't forget, there's some very clever defense attorneys out there that'll pick the state's case apart, but it's got to be constructed strongly, perfectly, legally, that the state are looking at going, oh yeah, I think we effed up here pretty badly, right? So you got to attack the forensic evidence. You got to attack the veracity of his statement. The guy was treated like an animal, worse than an animal, and he confessed, I think, 60 times. Hell, even I would have confessed to killing JFK and also anybody else just to get out of there, right?
That was my point of bringing up Buckland from UK. He confessed to a murder that he was a 17-year-old boy. And they arrested him because he worked in proximity. They brought him in, and he immediately confessed to raping and strangling a woman, or not a woman. She was a 15-year-old girl. And 3 years before her, there was another 15-year-old girl, and they were sold on him being the perpetrator of both crimes. And they submitted his DNA, and the DNA proved he didn't commit either one. Yeah. So the thing is and not only are seeking justice for Abby and Libby, you can think whatever you want about Richard Allen. Okay, the state did not prove their case. Number one, number two. But what this really boils down to is the fact that Richard Allen is not a millionaire. He's not a CEO of some major company. He was a real man living a real life and didn't own his home, owned 2 cars, had a wonderful wife, a wonderful life. And whenever the state decided to point the finger at him, he had no ability to save himself. No. And when you are an innocent person, and trust me, I'm not saying, I believe that Richard Allen is innocent, but I'm saying under any circumstance, if you are an innocent person, you have to ask yourself, how would you react under these circumstances? What do you say? How do you respond? I mean, there's no right or wrong answer. But in a situation like this with Richard Allen, he's a man who I believes I'm telling the truth. I'm going to let the law work for itself because I'm not guilty. And look what they have done.
It's almost the perfect example of what not to do. I'm not saying anything bad about Richard Allen in any way, shape, or form, but it's just, if a police officer asks you for your phone, it's not because he wants to have a friendly conversation with you. Make him ask a judge for it. Don't give him your phone. Don't let them search your house. Make them work for it.
And that's not a prelude to guilt. That is, I need to, right. You have to protect yourself. But Richard Allen, like many people in this country who have thought that honesty is the best policy. I have nothing to hide. I'm going to come forward and I'm going to talk about it. Then they realize it doesn't matter. You watch those interrogation videos, it doesn't matter what Richard Allen would have said or not said. When you find out that Doug Carter was called 2 days before this interview even took place and was told there was going to be an arrest made, nothing that Richard said on that day would have mattered.
Because they slapped all the photographs in front of him. They're going, this. And as soon as I saw the picture of the bullet, because our expert, a ballistic expert, said, "This is your bullet," and Richard's going, "Huh?
What?
I didn't even have a gun today." Because what the hell are you talking about? And of course, the officer is permitted to lie, right?
Mm-hmm.
So he was expecting Richard Allen to go, "Okay, mate, you got me. Yeah, put the cuffs on. I'm gone." And Richard Allen's looking at him and going, "Are you now accusing me of committing this murder?" He goes, "Oh, yeah, but I want you just to explain." Notice what the officer was doing.
I'm trying to help you.
Correct. He was trying to flip the tables. He's going, "Well, if you were in my position, what would you do?" As if Richard to say, "Yeah, I understand. Yeah, okay, keep accusing me. Keep on throwing all this shit at me." What Richard needed to do was to walk out.
He did.
Immediately.
Walk out. Walk out. Yeah. Yeah.
Hire a lawyer and never say a word to them again. Make them talk to your lawyer.
Yep, they did the same. They did the same thing to another case that I studied, Nick Hillary, right? He was an African American accused of murdering an 8-year-old, I think 8 or 12-year-old boy in his apartment. They couldn't find any of his DNA there, right? None whatsoever. And they got him on a false pretense to come to the police station to say, "Oh, we just want to have a talk about this and this and this," right? And so once they got him in the police station, they started to use the read technique and interrogate him. And in the end, he goes, "Oh, I'm not answering any more questions. I'm walking out." They prevented him from walking out, and they made him strip completely naked, photograph his body from top to bottom, and then they let him go in a hazmat suit. So even though Richard Allen wanted to walk out, the damage was already done. It was not good. All right. Do we have any more questions?
There are 108. You may have answered some of them, but they're starred, Amy. Can you access them, or do you want me to put them up?
I'll let you. We'll go through these and we'll put them up. We'll try to go through these quickly. So for this question, it is it common practice while using a rape kit to rule out DNA of a male if the victim lives with a male?
No.
That's not common practice.
No, their laboratory didn't test for female DNA because obviously the investigator said, "Well, we believe the perpetrator is a male," right? So that's a bizarre thing to do. But when they do a rape test kit, right, whatever results they find, right? Whatever results they report, they report. They're not going to say, "Oh, well, the girls live with a male. We're not going to do any testing with." No. They'll extract the DNA. Whatever profile comes up. Correct.
Okay, yeah, because the follow-up was, why even do a rape kit test if a victim lives with a male? So.
Yep, correct. No, that's got nothing to do with it. None whatsoever.
Okay, so this is from one of our Richard Allen researchers. She said, for Dr. Silkman, can you speak about advancements in technology for extracting DNA from rootless hairs? I'm asking because there were 3 hairs inconsistent with Abby and Libby that weren't tested.
Yeah, I basically answered that. There are techniques available to extract mitochondrial DNA from rootless hairs, but it is a specialized process. Now remember, your sample size that you're extracting is 1, because you'll have to treat each of the hairs separately. But there are forensic laboratories that specialize in this technique. Again, it's going to involve extraction, PCR, generation of a profile, because you can generate profiles from mitochondria, but they're different profiles. So yes, the technology definitely exists to extract mitochondrial DNA from rootless hairs. Correct. But it's not a routine thing. It's done in a specialized lab.
So, okay, so that will go into the next question, which is, if a more advanced lab with further advanced testing is available to obtain results, Why would this avenue not be pursued, and who makes that call?
Money. So in effect, you have to think, is this going to be probative? Is this going to be informative if I go down and down and down a pathway, right? So you can do these further testings, but they're going to be expensive. So their way of thinking is, and I think Stacey even said it, she goes, yeah, the chance of getting any genetic material from a hair is actually quite rare. It's not really a good idea. So they go, okay, we'll put it on the back burner and we won't test it. That's their way of thinking. Whereas the defense will say, no, give it to us, we'll do further testing. Correct.
So in a case like this, where you've spent 7 years doing an elaborate amount of investigations, whether across the board, is that less expensive than from Jump Street saying, let's test what we've got and get to the bottom of the source? Which one's more financially?
Well, the thing is that, and here's the problem, and it is a real problem. A crime lab doesn't just work on one case. So the staff in the laboratory, and this is the same with Wisconsin State Crime Laboratory too, where it was discovered they had hundreds or thousands of rape test kits untouched. No one analyzed them.
We have that same problem in Los Angeles.
Yep. So when they do a case, they go, well, we can't spend a year, 2 years, 3 years doing 1,000,001 tests. We'll do the test that will hopefully give us a result, and we'll take it from there. But having said that, the laboratory should keep all the forensic evidence just in case the defense wants to go, hey, look, I want to retest, I want to try this avenue. And even Stacey said, they're coming up with new techniques all the time, right? So preserve the biological evidence. As the techniques become more sensitive, cheaper, let's do it.
But
correct.
Now that we have a conviction,
no
one's gonna order those tests.
No.
So that leads into the second part of this question, which was for Andrea. It says, how would a defendant force the state? And I think you can speak on this because you've been through the process with Stephen Avery and the years and years and years of his legal arguments. How would a defendant force? This is when, like we have right now, Rick Allen being convicted. No one's going to be pursuing on the state's side of the fence. No one's going to be pursuing any kind of DNA testing because they believe they have their man. So how would a defendant force the state or their own legal team to get the advanced testing done?
Well, you saw what happened with Stephen Avery. But Kathleen Zellner requested all these forensic items for retesting. So she consulted with a professional DNA laboratory, forensic laboratory, and got all these items. And she tested the bullet and debunked it. Said, mate, this bullet did not strike bone. This bullet did not result in the death of Teresa Halbach. The bullet was the number one forensic item in the Avery and Dassey case that hung them both, because you've got the
just like this case
in the garage, and you're thinking, oh my God, the jury are thinking, well, if the bullet's got her DNA on her, they've got to be guilty. So then the state said, shit, we can't give Kathleen Zellner any more forensic evidence because she's going to potentially change the outcome of both trials. So they blocked her. They gave away the bones, I think 2011, and now they're blocking her from getting anything anymore because they know, oh my God, if they find another genomic profile on the RAV4 or another piece of forensic evidence that doesn't point to Stephen, doesn't point to Brendan, then the conclusions are wrong. In this case here, the defense attorney's got to say, all right, what can I request that's going to give me maximum impact? Clearly, you want to test the hairs, you want to get access to the clothing and say, all right, there's MVACing. Let's test the clothes. Let's test where you didn't test. Because remember, with MVAC, they were able to extract DNA from a rock that had been sitting in water. A rock. And they got DNA from it. And they found the perpetrator years down the track. So MVAC is a new technology. They obviously didn't try it. So the defense can come up with an argument. Well, your analyst said she found low levels of male DNA. Well, let's get a profile. If it's my client, he's screwed. But if it's somebody else, you need to explain how it got there. Why not? So ask for it. Ask for it.
But at the same time, it's more than likely down the road going to be a defense expense.
Or they could potentially do a GoFundMe.
Right. But I'm saying, like, in Indiana, which is one of the few states that provides state provided like post-conviction relief. They provide an attorney, but then even in this case, they were at the discretion of the judge approving what the defense needed as public defenders for experts, testing, that sort of thing.
Oh, yeah.
And I mean, I highly, there are so many people that follow this case.
Yes.
That I feel would benefit tremendously from going over. I mean, they might have watched Making a Murderer on Netflix, but it's like anything else. Like if you can listen to the Down the Hill podcast and get a brief overview of this case, but the actual ins and outs and the in-depths of this case are here. People who are in the intricate parts of, and there's a lot of great creators and there's a lot of great coverage on this case. But in the Stephen Avery case, we are now at the point where we are at the been there, done that.
Correct.
And Judge Flowers, aka Judge Flowers, is another Judge Gall who did anything and everything that she could do to roadblock Kathleen Towner and Stephen Avery from getting any kind of relief in the state court level. And the one thing that you have to understand, the Stephen Avery case is not just Stephen Avery convicted, but his nephew was also convicted.
Correct.
And they both went to
the same victim in two different circumstances, two different murder
scenarios. Two different trials with two different actual criminal scenarios. Like you would never watch both trials and realize that they are talking about the same murder because the scenario and the way each trial. So you've got two men convicted of the same crime that they were convicted on happening absolutely two different ways.
Correct.
So when you think that a case like this is not possible, I have serious news flashes for you. This is from the ground up a serious situation. And like I've said, we will find out how corrupt Indiana is when we get to the appellate court. When this direct appeal is filed, we will know based on their answer how corrupt this state is.
Correct. I mean, I haven't gone into the legal wranglings and danglings in this particular case, but the fact that the judge didn't permit any recording already tells me it's eyes off.
Yeah, eyes off. Eyes off.
The only way you're going to solve this case is to do what the Foul Play team did and other YouTubers. You need to deep dive. And trust me, when I mean deep dive, I'm talking super deep dives. I spent years going through all the forensic evidence with the Foul Play team, and I focused on the DNA forensic evidence because that's something I can talk about. and the shenanigans that we found, and also Kathleen's honor, the state want to shut it down. The state is going to shut down the Delphi murder case big time. Big time. We're going to hit a brick wall at every opportunity. So hence, whoever does a post-conviction appeal would have to consult with a current forensic scientist, criminologist, who would give advice to say, this is what we need to do. This is what we need to test. I could only do that if I see the original reports. That's why once you get those original reports and the testimonies, then we can go over it. Someone like Andrea, myself, we can go over it, read it, dissect it. We did the same thing with Daniel Holtzclaw. I did the same thing. Look at all the forensic evidence, have a look at the testimonies and see where the weak falls are. In this case, there are plenty. There's plenty. You can drive a truck through them.
So for the individuals, well, I know the direct appeal is already underway, and I think that there are people who are fighting to be able to start the post-conviction relief case, so that when we get it, I mean, we're looking at like 2 years before we get an answer on the direct appeal, and then if we get an answer not in the favor of going forward from the direct appeal, we then are starting all over again with the post-conviction relief. That's why some people feel that the post-conviction case needs to be started now. Would you be willing to consult with them?
I'm happy to discuss anything to do with DNA as long as I can get my hands on those original reports. Because like I said, for me, I need to go back to the original material. I do not look at 100 different YouTubers who talk about this, who talk about that. No, that's not how I work. I'm a researcher at heart. I'm blessed with the guys that we have on the Foul Play team. They are exactly the same. They're researchers, which means we go back to the original material, we dissect it, we read it. We did the entire Avery case. We read the entire Avery and Dassey case. We dissected it from pillar to post. I've done, Ken Kratz wrote a book on MAM 1 and MAM 2. I did, 43 or 63 podcasts on taking apart his book, right? That's how deep we dive. You got to do the same for this case. You got to dive deep and get rid of the noise. Get rid of the noise and focus on what the state got wrong and where you can potentially do further testing. I would definitely hit the ballistics, and I want to see all the DNA reports because I'm interested in seeing what Stacey didn't test and what Stacey didn't tell us. Right?
Okay. I'll work on that.
Yeah, that would be awesome.
I'll see if I can get that. That was the last question.
Yep, I'm sure there are plenty of people in chat who are very knowledgeable, who know a hell of a lot more about the case than what I do. Because like I said, I've only spent a couple of weeks on this case. I spent over 7 years on the Avery case and Dassey case every day. That's how obsessed I was. And I tell you what, I'm still learning new stuff.
So I'm just gonna scroll through chat. I'm gonna scroll through chat just a little bit.
But we do have, just so you don't forget, we do have over 100 questions starred. Like I said, you might have answered some of them. I just starred them all, and so
there's always a possibility we can do another podcast at some stage, and maybe go through the questions because we've done well over 3 hours, and oh my God, we've got nearly 400 people in chat. That's fantastic. Goes to show how people are very interested in the case. And again, I'm sorry, but I don't want to go into any type of rumors or speculation. My focus was strictly on the DNA forensics.
And you have been, you're amazing. I mean, I'm going to say it, you're amazing. The way that you detail your expertise is amazing. DNA is something that, a lot of people can get bored with if it's not their forte. If science is not their forte.
I love this stuff. I love this stuff.
Yeah, but you make it very understandable, and it makes you want to listen more, because there's all these questions about DNA, and no, you've been amazing, and I appreciate you so much.
And thank you for asking me to come on. I appreciate that.
Absolutely. So we do have a lot of questions, but we've already been on here for over 3 hours, and I've got a couple of text messages. Hopefully, I'll get with Dr. Silkman. Everybody, Foul Play on YouTube. Foul Play. If you type in Foul Play Stephen Avery, you'll find him immediately. Follow his channel. And he's got a lot of specialty videos on there about discussing DNA. But the Stephen Avery case, if you want a real life version of what Richard Allen and Kathy Allen are up against, the Stephen Avery case is a mirrored image of that. That is why it is. And that's why I told Cherie back when I started to believe that Rick Allen was the wrong guy. I said, and Cherie will tell you, I said, if he is found guilty, I will start covering the case. I have been following it, but my channel was designated, for Ashley Garth that you came on and spoke about. And we're still fighting that fight. But when he was found guilty, I said, okay, I'm going to cover it because, the people in this country need to understand what they're up against when it comes to the justice system and how easy it is for your entire life to be ripped out from underneath of you.
Destroyed.
So
and the reality, sorry for cutting in, Amy. The reality is it could be a lag time of between 10 and 20 years.
Yes. I know, and people think that, oh well, if he's not the right guy, he'll file an appeal and get out. It doesn't work that way. The justice system is not designed for, once you're convicted, to work in your favor. It's designed to work against you in a emphatical way.
Yep. Yeah, and like I said with Stephen, the reason why he got exonerated was because they found one pubic hair and it belonged to the perpetrator. That's an aha moment. And that's why the hairs are very important, because if one of the hairs belongs to potentially somebody else, how did it get there, right? And it's interesting that I don't find any of Richard Allen's hairs, as far as I'm aware. So again, they need to go back over the forensic evidence and check it again. Did they miss something, right? And that's why I recommend using MVAC, which is an incredibly powerful technique, because if cells are there, they'll vacuum them out, right? And they'll usually generate a profile, which is exactly what you want. Stephen got exonerated because of one pubic hair. Otherwise, we still would have been in prison, right? So that's hopefully what we can find in this particular case, something that will exonerate him. But the road is high. The road is steep to get there.
It is. It's an uphill battle. And if his family is watching, just know that, we're out here fighting, because
I don't know the guy. I don't know the family. I'm in Australia. I'm in the land down under. But what I can't stand is when I see injustice in anybody. Doesn't matter. Even if the guy's guilty, you got to make sure that you don't give the defense any opportunity to say, mate, he confessed under these bizarre circumstances that you won't find in a war camp in Nazi Germany, and you're treating him, who has a presumption of innocence, in that manner there. The guy's got depression, you're filling him up with drugs, and he confessed 60 times. So what? Even I would confess just to get out of there, right? So things have got to change, because channels like this, channels like Foul Play, you want to expose the shenanigans, and the crux of the problem is the read technique itself can solicit false confessions, right? So it's a disaster. Yet in the US, they still keep on doing it.
They do, among other things. Among other things.
Yeah, but I think in this particular case, sorry, Amy, in this particular case, you've got to attack the forensic evidence, right? Which is interesting because some of the jurors were asking Stacey, well, what do you do about this? How do you ensure this? What about that? What about contamination? Which is exactly what you need to do. Attack the forensic evidence.
Okay. Well, I know there are people are listening, and they're going to take your advice.
Well, I hope I've made some people think, and I'm not perfect myself, but anything to do with DNA, I can definitely explain it. Remember, I've taught thousands of students. I'm retired now. I taught thousands of students all techniques. I've taught people these techniques. We use PCR. We do all advanced molecular biology. I've done the whole lot. So, but I've got a passion for true crime, and it really drags you in. So, when you see cases like, and also the Sharon Reed, is it Reed?
Karen Reed. Yeah, Karen Reed, yeah.
Oh my God, that's another one.
Yeah. All at the same time. I mean, these are going on and Brian Koberger too.
Correct.
Yeah.
Correct. All DNA. It's all to do with DNA.
It's all DNA. Yep. So everybody in chat, I thank you so much for being here. I feel it's been a very educational live, and we've gotten a lot out of it. And Dr. Silkman, I think you'll be back.
You give me those transcripts, and I'll definitely be back.
Yeah.
Those transcripts and the reports, Stacey's reports, and are going to be something I can comment on to find the loopholes and ask further questions. But yeah, if you can grab them, might take 6 months, might take a year, but whatever you could do, I'd love to be back.
Okay. Well, thank you, everybody in chat. Have a good night. And everybody on the panel, just hang tight until I end the stream, and we'll chit-chat afterwards. So thank you, guys. And if you have any questions, post them on the replay, and maybe Dr. Silkman will, review the chat over the next couple of days and maybe answer some of your questions. So have a good night, guys. Thank you.
Everyone.
Thank you, everyone.